Hello, hello. It is. Okay. So hey, please take your seats. There's plenty of seats on this side if you wonder. Okay, so let's get going. Very much welcome to the DDLS annual conference. As you may wonder, I'm not Jan Ellenberg. He happens to be sick today, so I'll do this introduction of myself instead of him. And also welcome you to this meeting. It's really a pleasure to see almost a full sort of a auditorium here. We had 270 people registering to this event, so great to see the excitement here. This will be live broadcast on YouTube as well, so that you know. And if I have the next slide here, you have the program here. So we have a plenary session now, and we'll then have a coffee break and continue with the program. And with that, I will just sort of get going and welcome you all to the event and introduce the first speaker, who happens to be myself as well. Now, this is, by the way, the word cloud of the submitted abstracts. So this is you. So in case you wonder, and I think this is really good for us to think about what the community is doing, not just like what the people we just hired are working on. And surprisingly, a lot of proteomics, for instance, among the submitted abstracts. So great to see that activity. So DDLS program is all about the convergence of computational and life sciences, and that's the theme of what I will be talking about today as well. But before I get there, just to reflect the tremendously sort of exciting times that we are living, that we have the technology-driven revolution in life sciences. That's the one that typically SciLife Lab infrastructure takes care of. And then we have had data-driven revolution, and now on top of that comes the AI-driven revolution, and those are the two topics that the DDLS program is dealing with. And I can only show the number of publications on these topics on AI and biology and healthcare, is particularly the health topics that are really exponentially increasing. And this is probably the greatest testament to the fact as to how important the types of things that we do here are. We had both a Nobel Prize in physics to the fundamentals of deep learning, and then the Nobel Prize for chemistry this year awarded to AI-driven research. So this is really something to be proud of, and also shows how important it is what is happening here. And obviously a lot of the things that we will be discussing are about specific AI machine learning programs, which most of the research community is still dealing with. But increasingly we are also being able to make use of the generative AI capabilities that are being broadly available to us, and we should make use of to the maximum abilities. So, coming back to DDLS, and let's go to the year 2020 when the Wallenberg Foundation, particularly Göran Sandberg, was involved in making the decision on launching this program that brought us all here, and many of you are now hired through this program. And this was really insightful investment in its time, because we need to consider that this was way before the AI era. Before GPT, there was GPT-3 when Google Trends started to go upwards. So we started before, and obviously DDLS wasn't initially an AI-driven program. We obviously have to incorporate this today. And the latest addition to keep DDLS up to speed was then just announced last week, and many of our slides and statistics are not yet even updated with this, because Wallenberg Foundation donated another set of fellow positions and also funding for the AlphaCell program that Jan Ellenberg would have been describing maybe a bit more here. So this was now Sarah Mazur at Coave, and both Göran and Sarah are in the audience as well. So they are also keen to see how the program is getting going in this sense. So this is then the program with the updated figures here, 3.7 billion krona for the 11 partners that participate in this program across Sweden. Over the next 12 years, well, we may have nine-ish years left, and we have the 4 research areas involved in the DDLS that we talk about today as well. Now that we were given this fantastic opportunity, we have obviously had many strategic discussions and really difficult discussions as to, given the exponential developments and really the revolution in data-driven and AI-driven research, where are we going to be in 10 years? What are the capabilities that we should prepare for? Given that, for instance, anybody can write code and there will be AI agents and autonomous AI, collaborative AI, AGI and ASI, meaning the artificial superintelligence, and obviously the division between life scientists and computational scientists should come closer, particularly in the DDLS context, and obviously AI will become more and more better in whatever it is doing, while humans typically stay at a certain level, but we should work together to get most out of it. So the lecture theme today is, and I will have 80 slides to go through, so this will go at super speed. So state of the DDLS as of November 2023, which is about 3 years after we got really started. And I'll go through the DDLS strategies, and in highlighting what has been done, but maybe point out to the objective that DDLS aims to promote a change in how life science is practiced. This is a very broad impact that we want to have to the entire life science community, and as you know, that is very big. But one specific angle that I now start with is the attraction of scientific excellence in data-driven life science to Sweden. And this has been the DDLS Fellows program, and I spent quite a lot of time in introducing this part. There was a phase 1 recruitment. Phase 2 recruitment is almost complete. And now taking these things together, we have 9 positions in cell and molecular biology, 7 in evolution and biodiversity, 10 in epidemiology and biology or infection, and 13 in precision medicine and diagnostics. And that would be total 39, and 3 are still open. But as you saw the latest addition there, we have new positions coming up in to be filled in the years to come, and to ensure that we really keep up with the latest trends in the field. So this will bring it to a total of 50 DDLS fellows in a few years' time. Now what I did was to sort of take a little bit of a look as to what the DDLS fellows are working on. I just pasted their abstracts on AI and asked it to draw a diagram on what kinds of activities are underway. And you see that is distributed quite broadly across multiple keywords, and obviously almost this has been the #1 criterion when selecting people, that you are really data driven, are applying data intensive strategies and developing also methods in this field, not just using the technologies. Out of these research profiles, this is the fellows word cloud. And interestingly, I've noticed that the proteomics, as compared to the abstracts that were submitted, there's no word proteomics here, and genomics has become a very strong thing. So clearly the fellows are a bit more oriented in genomic themes as well as microbiome. So many people are sequencing microbes, and that's why these 2 keywords are there. Now comes the funny part then. So I asked the DDLS fellows to send me a couple of slides, and they came in all kinds of variations and shapes and colors and you name it. So I tried to just put them together, and all fellows don't sort of kill me at the end of the lecture, because I will just quickly have to go through this and maybe say a few words on what I think you are working on. So Wei-Yo Yang, I start from him. He's working on generative AI-powered virtual cell capability and automated microscopy to create pictures of cells. And this program is probably the closest link to the AI cell program that Wallenberg Foundation just funded for creating a model of a cell. Golnaz, a new SciLife Lab fellow, transfer learning for detecting regulatory modules in ovarian cancer and looking at drug-drug interactions. Björn Forsberg, another brand new fellow in the cell and molecular biology, working on cryo-EM images and particularly the dynamic molecular variation in them. Milka on cell membrane biophysics and looking at, for instance, lipid organization in membranes. Then moving on, and I did not get slides from all the fellows, so I'm not introducing all the fellows, but luckily most of the new fellows provided slides, so the emphasis is really on the new fellows. Oh, I should have said. So very quickly, back up. Wei-Yo Yang is not here. Golnaz, are you here? Just Wei-Yo. There, okay. So I wanted to introduce people at the same. Björn, Milka, not here, or I didn't see. So now you wave your hand. I focus on the slides. I'm not going to look at you, but please make yourself known so that the community knows you. So Tom van der Valk is using his sequencing strategy, where you can take a little bit of a drop of seawater and sequence organisms in a tagged way, and then doing the bioinformatics, where you get an overview picture of all of the things, be it microorganisms, fish, or even mammals and algae that have left a DNA fingerprint on the ocean. So great work for looking at biodiversity and ecosystem evolution. Tobias Anderman taking Earth samples for environmental DNA analysis and coupling that with satellite and other aerial imaging and making a joint analysis of those data sets. Kelly Swartz, complex traits investigations in plants. Then Mariana, a new fellow, understanding the past to predict the future of ecological networks, phylogenetic models, Bayesian interference, and so forth. And then new DDLS fellow Jennifer, who is working on mutational robustness and looking at the fitness of new mutations and their impact on the proteome. And then Lisandro, in a similar way, developing computational models on evolution and predicting on how evolution could take shape. Then moving on, and sorry I keep this pace, but hopefully you get some idea on the perspective of what is included in the program. Epidemiology and biology of infection, EBI. I'll start with Camila, who is the earlier generation of fellows who is working more on the immunology side, particularly on the gender differences in human immunity and its impact on our proteins and immunology. Astrid von Menzer deciphering pathogen dynamics in computational and virulence factors and so forth. Kaisa Thorel, a new fellow as well, with the focus on Helicobacter pylori and its role in host microbe interactions and also cancer causation and other impacts. Jinmei on human gut microbiota and evolution, and also its impact on health, such as type 2 diabetes. Then finally, the precision medicine and diagnostics fellows, and we have 13 of them, so there will be quite a few. But again, a focus on the recent fellows. This guy is not a recent fellow, Jacob, working on Alzheimer's and Parkinson's disease, in vivo imaging and proteomic analysis and their integration. Abhishek, also not a new fellow, working on the clonal hematopoiesis and its impact and significance on our health and also cancer risks. Wen Zong, working on multi-omics integration and its involvement in risk prediction and precision medicine. Avlant working on predicting cellular responses with interactome-based AI and predicting cellular responses to a variety of stimuli. In that sense, Muhammad working on cardiometabolic disease and progression of the disease, again based on multiomics networks. Gyali is working on pancreatic cancer from a multimodal approach standpoint in terms of predicting and influencing pancreatic cancer onset. Antonio working on tumor evolution models with genomic data and looking at lineage relationships and clonal evolution and so forth. Marcel Tarbier working on single cell and spatial data to look at sort of a heterogeneity and phenotypic switches. Kimmo Kartasala working on image analysis of human pathology samples and developing AI-based biomarkers and prognostication. And then Arian working on the tumor microbiome, making an interesting link from the tumor and precision medicine side to the microbial investigations. That's it. I did it. So now moving on to other things, and you have many of the fellows here, and obviously great to see the fellows also meet each others, because just based on looking at your profiles, you have a lot of things to collaborate amongst yourself, but also with the other people in the other research areas. Next generation scientists have to be also trained. They don't just come imported. So we have a DDLS research school that Olof and Petra will describe later in this program. So I'm not going to spend time on it, just introducing the key people in the research school who you should know in order to sort of deal with the research school issues at DDLS. And we are very much in the beginning in terms of this effort of training PhD students in the program. So this will grow and will be an important part of the program. Just to reflect on the fact that there's an ongoing search for a second year of searching for PhD student projects and then PhD students. So we first have a search for the supervisors and projects, and then these projects look for the PhD students. If I look at the statistics of last year's call, it is about 1% now out of all of the PhD students that applied were admitted. So if you are a PhD student in the program, you should be pretty proud of your success. Almost like a lottery win. Then out of the application from PEIs to host a student, about 11% were admitted or were funded. Obviously there's a great need for this type of funding, but just to show you the perspective that we hope that this is actually super high quality people, both the PEIs and the PhD students, because we have a lot of selection process in there. And obviously these people need training, and this is also a thing that need to be launched and beefed up during the program. And the SciLife Lab Training Hub has obviously had a lot of training efforts underway, and you can see from the website on what's cooking, and also if you want to host a seminar, you can get help and support from there. Next topic is data science and bioinformatics, sort of a services and expertise. And this is the part where we really try to have an impact on the whole research community, not just these internal researchers in a way. A big part of it is sort of something that happens at the SciLife Lab Data Center that is co-funded by SciLife Lab Infrastructure and the DDLS program. And obviously I don't have time to go through everybody and their roles in this. But point out to a few things that the data center is supporting the SciLife Lab data platform, and there have been many different data sets submitted to the data platform, and they seem to be actively looked at and used and downloaded for other people. And this is really one of the main points about the DDLS program as well, that we make the data better available. The data center is also involved in a lot of coordination of national and international collaborative initiatives in this space. And then we have the WABI program, Wallenberg Bioinformatics, which actually started 10 years ago. So this is actually a program that has been running for a long period of time, and many people are working in providing services on demanding bioinformatics issues to the scientific community. And this is Björn Nystedt's picture, which looks a bit messy on purpose because they just show how important they are and how many things they have been doing in the past 10 years in terms of publications and impact on the community. What is now DDLS specific is the launch of so-called data science nodes. That we have these 4 research areas where the fellows work, and we have 4 research areas where there are scientists who work to coordinate these activities. But we also wanted to have a data science node that is targeting and specializing to a particular area of activity, and this is linked to the data center, and it is linked to the WABI program as such. And these are now the people who have been hired at the various, you see the abbreviation, cell and molecular biology, precision medicine and diagnostic, and so forth. So this is now cooking and started its activities. And if you wonder what is cooking on, one element that I have time to go through is what is a common theme across them is to create so-called portals. And this pathogens portal is maybe the best example of things, and that also predates the launch of the DDLS program. But this has been now developed forward from the COVID portal to a pathogen portal, and it is really providing up-to-date information on, for instance, wastewater viruses and all kinds of publications from the community and data sets from the community, and keeps the community hopefully together and provides information to people who are working in that field. Similarly, there was a genome portal provided from the evolution and biodiversity side on all of the eukaryotic species that have been sequenced, and shows another type of portal. And a precision medicine portal isn't out yet, but will be announced in the future. So this will also hopefully sort of bring people together under this theme. This brings me to another thing, which is a little bit related to this theme, and that is that particularly in the AI era, we have an increasing need of bigger and bigger data sets, really global scale data sets that would be used for others and particularly for training AI models. And how do you get them? Obviously, you can use existing public data sets and global life science resources. Or we can launch dedicated big research projects in Sweden, such as the Human Protein Atlas or the Alpha Cell program that is being launched now. But you can't do that in all fields of science. Then you can obviously try to access healthcare and other types of data that isn't yet available, and this is super important. I'll get back to this at the end if I have time. You can use synthetic data, but that's really not a solution for most cases. But what we are trying to now coordinate is to maybe help a little bit the siloed nature of data sets in life science, and launch a pilot program on something which hasn't really been approved yet. So I'm talking about our plans for next year. Coordination of data creation and sharing across a group of PIs with similar interests. This is not a program to fund their joint work, but to fund them to come together, because we are spending in Sweden billions and billions of krona on PI-driven research, and often that is poorly connected to each other, and the data handling is not properly coordinated across PIs. So we hope that this call that hopefully comes out in the fall will be able to help the community and serve as an example on how data-driven research in the broader community could be better coordinated in the future. So final point that I have a few more slides, and then I quickly summarize at the end. Collaboration between the different Wallenberg funded initiatives was part of the initial plan, and obviously our big brother has always been WASP, the Wallenberg Autonomous Systems AI and Software program, which has funding to collaborate with DDLS, and DDLS has funding to collaborate with WASP. And also we have the WASP HS program, which is humanities and social services, and I'll say a few words on that as well. We have quite a few projects running, about 30 collaborative projects between the data science, between the WASP and DDLS already. And what is actually now open or just closed is a program to spend 90 million krona on 3 projects of a bigger nature for 5 years. And this is where we have 24 applications now received, which will be then pre-screened and internationally evaluated. This is something that I wanted to point out, one particular thing also in regards to these service models that we just talked about. And that convergence in the future will probably take us to another level of collaboration. So typically we hear that life scientists do not understand data science. And if you talk to the data scientists, to the life scientists, they said data scientists don't understand life science. And I think that we cannot rely forever on a model where professional data scientists analyze the data for biologists. So therefore, I think that we can actually have multiple examples, like biologists have learned to program and have learned to analyze their own data with the help of AI. This is really something that we should propagate because that's more sustainable. But also the other way around, that AI can help the data scientists to understand the complexities of biology, which they were often not trained to fully understand. I think we should make better use of that. And I think this kind of bridging is really an important function of DDLS. And that's partly the type of future that we want to see, people who can handle the both parts, maybe with the help of a little AI, and that's okay. So finally, a couple of points on partnerships and policy actions in DDLS, but before I do that, I have often had in my presentation a list of concrete examples where AI has taken us in the field of biology, medicine, and so forth. I don't have time to go through that, but the important point here is that many of them are Nature and Science and New England Journal of Publication. So really serious science proving and showing the tremendous utility of AI across the wide spectrum of life sciences. I'll just point out one very simple thing, because there are many people who doubt whether these generative models have any kind of utility. Even today, when the GPT-4 model isn't in any way trained to be trained with medical data, it can diagnose complex clinical cases on paper better than general practitioners in Denmark. This was a Danish study. So either AI is very good or Danish GPs are not very good. But I think this was an impressive study published in New England Journal of Medicine. I don't have time to go through these sets of examples, but there are many. And as you know, AI in a number of ways, depending on how you measure it, is achieving human level performance. But despite of that, I often feel, and this is my empirical plot, this is not based on statistics, there's a minority of people who really embrace these things. I'm certainly one of them, and maybe many people here are such as well. But for many of these models, they just say this indifferent, pessimistic, doubtful, or that's what basically scientists are meant to be, critical, and that's fine and good. And then others are negative towards the developments. But I think there are already so many use cases, and AI is already vastly superior to humans in terms of the memory capabilities, remembering everything basically, the speed of the tasks, and the multidisciplinary knowledge. So most people start using GPT that they ask their favorite protein in their favorite disease and see what the AI can tell them. And surprise, surprise, it didn't tell anything that it didn't know already. But the point is to ask and be able to combine knowledge from all across the vast sphere. And this actually takes me to this point that we are supposed to do in the program, is to look at the ethical, legal, and social implications. I asked AI to list global ELSI challenges, and particularly Swedish challenges that the DDLS program could play a role with. That's my AI colleague at work here. And here I actually make the bridge analogy again, because I think this is really where life science and the WASP-HS type of ELSI questions could be looked at with the help of AI, because AI can help with that multidisciplinary bridging. the AI is at your home and you can learn and study and you don't need to talk to a live lawyer or a live ethical expert all the time. This is tremendously useful. But we are not going to outsource the AI, the ethical and human and social implications to AI, but this brings a segue to our joint conference that we recently had with WASP and WASP-HS programs to discuss these capabilities, what the three programs could do together. And we had a call together with the DDLS community and WASP-HS, that's the humanities and social sciences, and we have 26 applications. So now all of a sudden, the community outside of the life science community is getting interested in the significant challenges that we face here, and I hope there's a lot of exciting things coming out of it, and hope that you can also participate in these types of activities. This takes me to my final thing, that one theme of this conference is open data. And obviously, this is something that we deeply care for and want to propagate in every possible way. But where this is seldom possible is in the healthcare setting, where we need to have federated or other safe solutions of actually taking care of the analysis. And increasingly, sort of this is actually what we talked with the fellows just recently, not just bring health data to the compute, but bring the compute back to the health data and secure settings, and then connecting this thing. That's what the swarm learning on the right side does very effectively, kind of an edge computing capability. So I think in the future we need to do something about precision medicine, and diagnostic is a hugely part of the DDLS program, and we have and continue challenges in this scope. My last slide is this one. Just to show you that there's a lot of activities underway at the DDLS program. Very important part is all these meetings, and for you to meet each other and know about what's cooking in here. And this is not even a comprehensive list, but you see the activities, and I hope you pay attention to the SciLife Lab website, because that's where you see all of the events that are going to happen and that will go on. And with it, I will stop and not take any questions from you, because we are late, and I want to introduce the next speaker. I hope we have plenty of time to discuss, and please come to me and sort of discuss whatever element you like, but we will more or less discuss every aspect that I showed today in specific sessions later on today. So with that, so the next speaker and our plenary speaker today is Samuel Kaski. And maybe I should say, because I happen to know Samuel from before, I should say that I did not ask or come up with him to be invited. It was my fellow DDLS fellows who suggested him to be as a speaker. So Samuel is a long-time AI expert and machine learning expert. So he is currently a professor at Aalto University in computer sciences and runs the Finnish Center of Excellence for AI. And he is also sharing his time as a professor of computational sciences at Manchester University, where he is involved in a Center for AI Fundamentals. And he will talk to us about collaborative AI for science. So welcome, Samuel.
Thank you, Olli, and I'm very happy to hear you. I can hear myself really loud and clear, so hopefully you're not quite as so well. So I am going to talk about two things that I am excited about, and then a bit on what the two communities that Olli already mentioned are about, in the interest that if there are I saw lots of interesting connections between what the fellows here are doing and what we are doing. If there is anything you spot of interest, reach out to me or directly the people there. I will need to leave today. I have a flight to catch, but I will be here for the afternoon, and of course feel free to reach to me afterwards. So I'll start with the two obligatory AI jokes. So I have 2 slides that are AI generated. Olli is much more sophisticated than I am. I spotted lots of stuff in most of the pages. So the first one is about, for those of you who are still skeptical on what you can do with generative models, I asked advice on having a painting to hang on the wall or show on a presentation, a quotation on AI and maybe some critical elements. So trust, but verify, ensuring AI accuracy for reliable results or something like that. The second one with the same gist. And now this was actually slightly surprising to me because people have been criticizing that these language models don't have a sense of humor. We can of course argue on how much we like this humor, but at least there is some things which may be hardwired. I certainly didn't hardwire these with prompt engineering. So either they have been fine-tuned for exactly these kinds of occasions, or then they just picked up some of people's jokes from social media. But yeah, this main spirit I do buy. So I asked a colleague who is speaking a lot to young students or at schools, who often needs to introduce to kids on what kind of an attitude would you take to generative models or large language models. So it's like a little sister or brother. If you ask them to do your homework, they would be really eager and do lots of stuff, but you probably will want to check what comes out. So I give a couple of examples that are of different nature and that are not mine. So this is my colleague from Imperial College, Aldo Faisal, who is a Turing AI fellow as I am. I have the senior title. He is more promising and upcoming than I am. So in 2018, he published this paper where the gist was that you have an AI reinforcement learning assistant that observes what clinicians did in making the decisions and learns the policies. And then it turns out that the AI assistant is actually much more accurate in making the decisions than the clinicians are after the learning. And at that point, these kinds of news were really surprising. How is it possible that AI is so able to solve this problem? We always had headlines of this type. Or that can AI surpass our capabilities in task X and so on, which nowadays, of course, are not news at all. The real issue is what problems do we want to delegate fully to automatic solutions, where we take the risk that if we didn't specify all of the important factors, of course the system will neglect those, and that is the risk we would be taking. Olli already brought up the Nobel Prizes. I don't need to say anything more about this. So but I'll tell one anecdote. So my PhD supervisor, Teuvo Kohonen, was he remembered to, when I was doing my PhD thesis, he remembered to tell several times pointedly that he published the Hopfield network of the second kind a couple of years before John Hopfield, exactly the work that he got the Nobel Prize for. But Teuvo is not alive anymore, so now at the moment either he would be very happy or extremely bitter. About collaboration. So I'm talking about collaboration now in two senses, and this is the first sense, and this matches partially to what Olli presented earlier, and we certainly didn't synchronize these in any way. So I think the partially is exactly the right word here. So at the moment, it seems I'm talking about slightly different problem now. So I'm fully on board of what Olli said, that nowadays everybody should learn to program irrespective of their field to some extent, and everybody who programs should use generative AI to assist in that, but with the philosophy that I had in the two first slides, right? So now what I'm talking about here is trying to do something new which requires some solutions which cannot be bought at the moment, also on the AI side. Then at the moment, the main dominating collaboration mode seems to be that there is an AI research group or a few AI researchers trying very hard to come up with the new solutions together with a researcher in biology, for instance, or with a company. So it would be a division in that company or then another company. And of course, kind of looking from afar, this can't be more efficient than if we would have, these are after all kind of domain agnostic techniques. So it can't be more efficient that we only have pointwise collaborations than if that we would have collaborations that cut across several different teams according to what is most beneficial. Of course, there is also the other mode where there is a big company, typically big tech company, who dominates completely a certain kind of solution. But that then stagnates everybody to the agenda of that company, of course. So that cannot be the only solution either, unless that company happens to be very good. But I wouldn't want to risk that, giving a monopoly to anybody. So there is another solution, and that also touches to what Olli was saying, the open data, open research in certain ways, in controlled ways. So some companies and research groups have realized that there is actually a resource, there is a free resource if you use it sensibly. So the free resource is that if you release a data set, a problem, and if you do that slightly more professionally even and release a gym, that's called a gym in AI research, something where the AI researchers can use as a benchmark to try their skills and compete with their peers, then they will immediately flock into trying to solve the problem. And typically nobody needs to pay anything. And why they do this is that they actually would want to contribute to solving real problems, important, interesting problems, instead of working on the really boring ones that big tech comes up with at the moment. So now the big question, going back to that one, so the big question is, this actually is really good for science if we can utilize this really well. So what can we do to make this easier, both for a biology group or a group in any field and the AI researchers then to try their skills on that problem? So now if we look really hard, you may disagree with this in your specific problems, but I'd say that squint a bit and you will see this loop of design, build, test, learn cycle in the research process somewhere. It has different names here, the design, make, test, analysis, cycle and so on. And what I simply mean is the age-old thing from the beginning of empirical science of making based on a hypothesis, making an experiment, testing, analyzing the results and revising the hypothesis and so on. The same cycle occurs also in engineering design, for instance, arguably in any design task. Then you would make an artifact, you would test it, analyze the result, improve your design and so on, or product development. So what only has changed, or mainly has changed since the beginning of empirical science, is that nowadays this loop is typically run first based on simulations. So virtually in the sense of running them on the models and simulations many times, and then only going to the real world and testing the result much more seldom. For the reason that in drug design, for instance, of course, clinical experiments would be laborious, costly, take time, and even unethical if we run them unnecessarily. So now the state of the world looking with these glasses on is that every single field is running this kind of a loop in one variant or the other, and they typically have completely independent toolboxes. Each field has their own toolbox for Bayesian optimization, for instance, which is one approach with which you can automate this kind of cycle. So there's an obvious scale advantage available. So if we now generously call the methods that are applicable across these different fields AI methods, then developing them only once or developing better ones would be an obviously better solution than having separate solutions in each field. What I am particularly interested in my research nowadays is that could we, now that everything is digital, we don't need to build robots to help researchers in different fields. We can do that, but we don't necessarily need to. We can operate on simulations. could we have something that it has the same scale advantage, which would take one more step forward in helping the researchers in what they are trying to do. And that's what I would call AI assistance or collaborative AI in the title of the talk. So what is needed in order to do this virtual laboratory? By the way, it can be virtual reality, but it doesn't need to be. That's not what I mean. I mean simulation-based. Virtual means simulation-based. What is needed in order to do this? In essence, digital twins, which would be data-driven models coupled with the data sources of the processes that we are studying, the biological processes, for instance, of the instruments that researchers are accustomed of using, which makes the results interpretable, and also the AI assistants. And now I would then argue that we need to have some kind of user models. I'll get back to this. If I want to build an AI that is able to help users, researchers better, it needs to have some kind of a user model. So that's very high-flying up to now. Let's make this slightly more concrete. So what could be the simplest example of this type look like? So that now the setting is that there is a design task, in this case drug design, and a researcher who is not able to pre-specify exactly the types of molecules, any scoring function for the molecules that would capture all of the aspects that they want. If you are able to do that, then you can automate all of this and everything is done. Typically, at least I would be very bad at that. I would be able to partially specify my scoring function, but not fully. But I might be able to tell of the molecules which ones are closer to what I actually wanted, which ones are not at all what I wanted, if somebody gives them to me. So if this holds what I now said, now we can build an AI system that produces a set of candidate molecules, shows them to a designer, who then checks the ones that seem good, throws the others out, and then a system which tries to improve the scoring function, produce the next set of candidates and iterate in this way. This would be a simple example how this can operate, and this we certainly can already build. If this is too hand-wavy, too heuristic, much of what I now said can be rigorously described as a new kind of interactive prior elicitation for the Bayesians among us. Now I expect many are thinking that, well, yeah, great idea, but I mean, what if, can we really publish a paper where we had some experts giving some feedback without exactly explicating what was needed, and can we trust these results? Which is of course a very good question. So we asked a couple of years back, the question that now in a setting where you can do some empirical measurements, but they are typically more costly, but you can, I mean you meaning the AI now, can also ask an expert, but doesn't know how reliable the expert is, how much the expert knows. So can we make the AI's algorithm robust against having an expert who actually doesn't know? And the answer is that of course we can, right? So it only needs to maintain two models. One is only computed on the empirical data, the other one is computed on the combination in the best possible way, and it is maintaining a statistical criterion on how much the two models need to diverge before the risk is intolerable in a well-specified statistical sense. And this can be done. If any of you are interested in contributing to these kinds of things, this virtual laboratory movement, what is needed is software, some fundamental work, and then practical cases. And all of this is open and contributors are very welcome. So now after this motivation of collaboration, why we need collaboration, why I think it is beneficial, things might seem simple. Now the difficulty comes from that we humans are not just data sources. So far that's what I essentially assumed, so that we passively just give feedback and as truthful as we can. So in order to try to capture that this actually becomes very complicated very easily in very simple problems. We designed a game. If you know Bayesian optimization, this is essentially Bayesian optimization that the AI tries to do where the environment is the user. In other words, now the AI tries to maximize a function, so it needs to choose a point x on the horizontal axis where the function, which is the y-axis, has the highest value, but it doesn't observe the function, so it's a black box optimization task. But it can ask the user. What it has now done here is that it has asked the user at 2 points, you see the circles there, and now it's asking at the third point xq, and the user sees the function, and the user's task is to help the AI find the optimum, find the maximal point of the function. And the user can just give a value by moving the slider that you see here. So now if I'm playing this game, I would be assuming that the AI has some representation of the function, but it has lots of uncertainty. I would assume probably some continuity by Occam's razor, and I would assume that it's more uncertain where it hasn't observed anything yet. This would seem sensible, and I wouldn't explicate these, I would just do this intuitively, but that's probably what I would, if I need to explicate it, probably something like that I would be doing. Now if the AI asks me the value at xq, now if I just give the value, I would probably think that, oh, that now this is not going really well, because now it's very unlikely in the AI's point of view that the optimum is where it really is because of the continuity of the function, right, as you see there. It's outside, the optimum is outside the confidence bounds. But now nobody told that I need to actually give the function value. If I, on the other hand, if I would answer like this, after that the AI would actually have a gist. It would be very close to knowing where the function value is, and I might be able to help the AI quicker. Now this is just me, so my hypothesis on how I would answer this. I did actually play this game, but I wasn't one of the one of the study subjects in the experiment that we then ran. So we actually had several users do this. And as a function of the iterations, which is the x-axis here, the users were able to help the AI find the optimum significantly quicker than if we would have given the right function values. Nothing was mentioned about lying or not. Nothing was mentioned besides that the goal is just to help the AI find the optimum. And it turns out that the more the users lied, the more they were able to help. And now lying, of course, here is a strong word, right? The goal was to help AI find the optimal function point. And of course, you can interpret giving a non-correct answer in a narrow sense by giving a value that is an approximation that is maximally useful for the next step that the AI needs to take. Okay, why did I give this example? Is if we now have an AI which would take the user's answers literally, so that is the best approximation of the function value right now, it would be really misleading, and it would actually hurt the AI's capability of finding the optimum, right? On the other hand, if the AI has a so-called theory of mind, right? So if it has a model that the user probably has a model of the AI, with that model it can interpret what the user is doing and actually can find the optimum quicker. So users are not just data sources, means that we typically have an agenda. We want to achieve something, and our actions are only really understandable given the agenda. Even in these kinds of ultra simple problems, which makes building these kinds of AI assistants slightly harder. But on the other hand, for AI researchers such as myself, that makes this much more interesting. Okay, so what is needed for this kind of AI assistance? I already hinted, we need some kind of a user model. So instead of just thinking on how do we automate, so if we originally had a researcher who is doing some experimental designs and then running experiments on the world, making some observations, if we want to assist the expert in other ways than just automate, we need to have something more. So we need to have assistants. This is a desktop assistant, which is not AI generated, by the way. But I did ask also the AI on give me images about AI assistant helping scientists. And now I got two different types of images. I was actually expecting to see the images on the left. How I interpret this is that there is some kind of a genie of the bottle which is trying to help the scientist who is looking at the microscope better understand what there actually is in the microscope, which would probably make sense. But then we get these nice images on the right. And of course I'm showing this because it happens to fit really nicely to my story. So at least I can potentially see their AI looking at a model of another AI or a user. And it absolutely needs to have exactly that in order to be able to help. So we need to have an AI which, in addition to having the world model, so-called world model of the problem that is being solved, like a biology model, it needs to have a model of the user it is trying to help. And with that, it can judge what would actually be helpful for that person right now. And that's kind of a theory of mind. So attributing a theory of another being, which is the essential property that we have, or characteristic that we have, which enables us to communicate efficiently. Otherwise, it would be really tedious. Okay, we wrote a paper meant to be a tutorial to these kinds of things for design tasks, where the assumption is that we have a candidate design and we are doing sequential decision making, trying to judge how to change this design so that it would have more of the positive characteristics and less of the negative ones, under the condition that we would not be able to specify the goal function, objective function or utility function that we have, but we would be able to tell apart better solutions from worse solutions. If we now have an AI assistant that has some model of our sequential decision-making process or planning process, it can help us by making some shortcuts because it will see more data than we see. One instance would be drug design. Another instance would be designing a day trip to Paris in this case. So where I have a few hours to spend in Paris, I know roughly what kinds of things I enjoy, but it would be completely impossible for me to give a utility function on those. On the other hand, when I see a suggestion, I would quickly be able to tell whether that's better or worse. And I would have some budget constraints, time constraints. And now the assistant in this case would be proposing for the current route, proposing changes. Maybe you could fit in Eiffel Tower if you haven't been there. It would only cost you so and so many minutes because you happen to be nearby and so on. So now in a simulated case on the right, as a function of how many recommendations you need to take and how highly you would value the resulting route, if you do an AI assistant in ways that I'm skipping all the details of, it would be able to help you optimize the route much quicker than if you would operate without the AI assistant, and much quicker than the alternative approaches, which I will also skip for now. A really difficult problem then comes on, this seems sensible, but how on earth would we build a model of human decision making or planning capability? Sounds like a daunting task. Fortunately, there's a principle that helps us get started and actually rather far. Algorithm is harder to devise, but in certain cases we can do that. I call this no shooting in the foot principle. So it's more likely or much less likely that we would intentionally do something that is against our utility function. But obviously we are limited beings, starting from not having infinite amount of time in making the decisions. In economics, often the the theorem starts by let's assume a rational decision maker. And now that I understand what that means is, of course, there is never a rational decision maker, because it means that you have infinite resources and infinite time. Obviously, that's okay. Let me not say anything bad about economists, but what does make sense is that we have constraints. Some come from the tasks, some are kind of side issues like how much time you have available, some are cognitive constraints, some come from the task in other ways. Some can be parameterized, some not. If they can be parameterized, they can be learned from observations of behavior. And now this gets us actually started. So given the constraints, the forward solution, given a utility function and constraints, would be we can compute that with reinforcement learning. And in principle, the reverse solution of what is the goal of the user could then be computed with inverse reinforcement learning. In principle. In practice, of course, there's lots of difficulties, but at least now we have a starting point. Now we know how to think about this, which is a big step. One more thing I need to mention about this, when I talked about the theory of mind. So now we need to decide kind of how far does the AI think of this. So it can think that the user has a goal. So that's the level 1 there, top right. So there's this maze in the head with the flag showing the goal. That means that there is the user is doing planning and it has a goal. AI assumes that the user is doing planning and has a goal. That's exactly the kind of example, the Bayesian optimization example I showed. Bottom left, the user is having a goal and it knows that AI has a goal. Or actually, this is what the Bayesian optimization was. So that's exactly what the AI would be assuming in that case. And now you can build a hierarchy of this. And probably in adversary strategic decision-making cases, you probably need to go further in the hierarchy. But I would argue that with level 2, at least, that's good enough for any of my decision-making. I would never go deeper than that when I do something in practice. So if you are interested in these, we wrote a paper for modelers especially, who it turned out that most of my students by default would assume that anything related to human-computer interaction is a completely side issue and very hand-wavy and not interesting. Then the students of my colleagues who are doing human-computer interaction, they think that machine learning, on the other hand, is too focused on nuts and bolts and not focusing on the really important questions. Then my Bayesian colleagues would be thinking that, come on, isn't the whole idea of science is to take the observer out of the loop? So why are you talking about the observers? This is really non-rigorous and I want nothing to do with it. So now we wrote this to clarify, and with a simple example. So let's assume young Newton observing the apple falling from the tree, and then let's assume young Newton growing up on a space station. And if we try to build AI assistants helping young Newton develop theories of gravity, what should it do? What should it take into account? And now actually we need to do the kind of next generation modeling, where we actually need to put the user back into the loop. And now, of course, all the old physicists turn in their grave. But this is actually rigorous. So you need to put the user in the loop so that you can treat the user as a nuisance variable and integrate over it is the right solution. That's the next generation physics. Okay, so now all principles are here. We just need to compute. Now the bad news is that the compute actually will take a lot of time. So if we do the brute force solution, which would be called simulation-based inference, approximate Bayesian computation, or likelihood-free inference, where there is a reinforcement learning problem in the inner loop, it will take hours, if not days, for a very simple user model on menu search. Fortunately, at the moment, with the advances of neural computation, there is a solution available which is called amortization or pre-computation. So based on simulations, you would create a lot of training data, learn an approximator, and then apply that approximator, which is very fast online after you have computed it. even fast enough for real time. It turns out that it only takes a few milliseconds to compute online for an assistant that would help the user save time up to 50%, which is about the same percentage as the knowing what the user really wants to do. So it seems promising. So I have a couple of minutes left, I believe. Olli showed 5 minutes ago. So a couple of examples. So this has all been domain agnostic about the collaboration. Of course, I am interested in biology medicine as well. So I am also working on models of biology medicine. A few examples. A foundation model population-wide, taking into account also the pedigree structure in doing predictions, disease predictions. Another one, single-arm clinical trials, trying to avoid the problem that we would unnecessarily, in randomized controlled trials, assign additional people to the control group, and as a result they would not get treated. Because if they haven't been treated in another experiment before, of course they would exactly in the same way not be treated in my new experiment. If we can do these matchings, we can save a lot of suffering, hopefully. And that can be done with certain kind of models. Third example, if we have heterogeneous diseases with unknown subtypes which have different causal mechanisms, by having suitable causal inference proxies, we can identify them and do much better predictions. For the cases where data is distributed like to different healthcare providers in UK, and if we don't have a control of these different providers so that we could have federated solutions, an alternative solution is to release synthetic data from each of these, so that which would be differentially private, so that everybody would learn from the synthetic data of the other partners. And as a result, most of the healthcare providers actually would benefit a lot. The biggest ones not so much, but they wouldn't lose either. And then this I will skip, just mentioning that combining different foundation models, which are kind of experts of different fields for the purpose of solving more complex problems, begins to be possible. And then very last, I'll mention briefly something about the centers which I'm involved with. The Finnish Center for Artificial Intelligence, 70 professors in AI. The numbers you can see there, lots of collaborators, and many are working in healthcare, for instance, would probably be interested in collaboration. Center for AI Fundamentals in Manchester is a startup center. So when I went there, they gave me 5 professors to allocate as a startup package, and now they have already hired 9, I think, because we got so very good candidates working on these topics across applications in different fields. So the fundamentals here means that we really are taking the machine learning seriously. And last thing I wanted to mention is European Laboratory of Learning and Intelligent Systems, ELLIS. And why I'm mentioning this is that ELLIS doesn't have much activity in Finland, in Sweden, and I think you guys are missing out. So ELLIS is at the moment it's 41 units in 16 countries. Out of the 20 best European universities, 18 have an ELLIS unit. And some of these are now being expanded to the next generation units in attractiveness and size. And we are at the very last stretches in launching one in Finland. It will be the second one after the first one that is in Tübingen, Germany. And the Ellis Institute Finland will certainly be very happy to collaborate with the excellent fellows of DDLS. Thank you very much.
Okay, thank you so much, Samuel. Really fantastic work and lots of sort of links with our work. So we have some time for discussion. People will be able to hand out the microphone. Maybe I start while, if I don't see a hand popping up right now. Oh, there, okay.
Fascinating talk. You basically indicated that there should be some level of lying to an AI in order to inform it maximally, and whether or not there should be some sort of hedging that in how much it trusts that information. But that was also in a system where it seemed like you had limited information to actually convey the full complexity of the problem. Is this sort of something general where you can sort of say how much, based on how much missing information there is, how much a learning model should hedge or sort of attach uncertainty to observations? And it seems to be related to sort of strategies in optimizing learning to some extent, but I couldn't gauge whether or not this was a general thing or specific to limited information cases.
So I think what is necessary is that there are degrees of freedom that you can use, right? So then you could use by doing planning, and by doing planning where you take into account the other player, you will be able to give more useful information if it is a collaborative game. And this was all collaborative, not adversarial. So yeah, I suppose game theoretically those would all be limited information games. I think you are right. And there certainly are general properties. I don't think people have studied them very much with the theory of mind in the loop. It gets very difficult very quickly.
Okay. Well, maybe in the meantime, I'll take one. In one of my sort of slides that I usually go through, there is one of these published studies where people had sort of worked again with the general practitioner solving the medical questions and how much they can solve it alone, how much can AI solve, and then if you put the physician and the AI together, the result is worse than with AI alone. So how do you then see this kind of being taken forward? Because the very practical solution is these medical questions and decisions and so forth. So how do you then go towards implementation, testing, and sort of proving the right way forward?
Right. So now, yeah. So I bet that different studies will be very different in how they were done so far. My guess is that now there would be some ready-made AI system applied in a certain way without considering how it works well with the human, which would be the reason why they don't work well together.
There's also the simple term that people are opinionated, that they jump to conclusions in the medical field.
All right, fair enough. And now if you incite that bias with the AI, okay, I can see. Yes. So the bias is certainly, I mean, there's nothing you can do if somebody insists of being biased, right? But if somebody wants to get over the biases, then these methods that would actually infer the constraints that the user has will be able to help, right? So, and that we can already show now. But then how to go forward in actually building systems and showing that they operate better, that's a complicated issue, obviously. So the incentives need to be right. There's lots of practical work. There needs to be lots of HCI, human-computer interaction expertise. There needs to be lots of AI expertise. The current solutions are not geared towards that. And then there needs to be a lot of engineering on the how to place that whole thing in the practical practices of the GP.
Okay, anything else? So a very quiet group today.
Maybe I was so comprehensive that there's nothing.
So, there.
Thanks for a brilliant talk. I just wonder that when you mentioned the principle, don't shoot yourself on your foot, I wonder how generalized this principle can be applied to other fields, and whether there's a local weighted minimal to search for or global weighted minimal to search for. Maybe you elaborate a little bit more than I can understand.
Yeah. So specifically, it means that it makes sense for the AI to assume that the user is more likely to operate in ways that match to the objective function. So if you have a goal, then it's more likely that you are doing things that actually advance that goal. It's very hard for me to find an argument against that. Another version that I would call is that it's very unlikely that people would shoot themselves in the foot unless they think it's good for them, which would mean that then your utility function actually is something else. But I'm not sure how to answer your question. It's very, at least equally general than my statement.
Thanks so much for excellent talk. I will just speak.
No, it is on.
Is it on? Okay. So I guess my question also very related. So in biology, often many problems don't have this known F function, right? Which you just demonstrated, and basically all the biological experiments often are done irrationally, without kind of knowing what it will uncover. So in a way, what is truly missing is probably some kind of generalized active learning framework for experiment planning, or basically giving not the feedback to the AI system, but other way around, to also giving, let's say, a data, right? What would be the best ways to do experiments that would uncover more knowledge? Do you have kind of any maybe thoughts about that? And just not that we need to teach AI, but often for many problems we can make generative models such a way that they know many more things than what we know, but what is missing, that okay, they have some uncertainty in some parts that needs to be learned, and if it would be nice that you could somehow have a kind of a general reinforcement type of problem that would say, okay, you need to do this and this and that experiment, then I will be much smarter, or something like that.
Yeah, so there is no need to assume that the goal would somehow be very sharp, right? It's a perfect goal to say, give me most information you can about the thing I am studying right now. But now there is a theorem that says no free lunch. So you can't get anything out unless you are willing to commit to something, right? So if you try to do just information gathering without any assumptions, then it really is random. And then just completely, the more random can the strategy be, the better. We already know that. But it could be, for instance, given a hypothetical model, kind of tell everything, kind of do the measurements that help me best know, or whatever there is to know regarding this model and neighboring models, for instance, which is ill-defined but would be a direction to give. So any way of giving a direction. And I agree, this is much more interesting than assuming that there is a sharp objective function that we just need to uncover, and once we found it, then problem solved. Very few real problems probably are like that. At the very least, it would be a moving target. But yeah, sorry, I don't have a kind of, in a nutshell, answer to that one. That's a difficult one.
Okay, anything else? I don't see anything, so I think it is about time for coffee. So let's thank Samuel once more time. And please be back after coffee for the fellow session at 1:15. Thank you.
I'm trying to get an eye contact, so please take your seats, everyone. I love hearing the bus, but we do need to get started for the next session. So please hurry up and take your seats, please. So it really looks like everybody's been enjoying the coffee and a lot of discussion, but let's get started so that we can keep the time of our next session. So, sorry?
Really?
Okay, so would you please increase the mic volume?
Oh, this.
If that's possible. Okay, so please take your seats, everyone. All right, I got a thumbs up for the sound in the back. So most welcome to the second session of this annual DDLS conference. And I really enjoy hearing all the buzz in the room, but now we need to listen up because we have 3 very interesting talks from both DDLS fellows and as a former SciLife Lab fellow. And first up, we have Laura Carroll here, who is a DDLS fellow at the Umeå University. And she will be talking about data-driven methods for microbial secondary metabolite discovery. Most welcome, Laura. We look forward to your talk.
Thank you so much. Hopefully you guys can all hear me. Thanks for the super nice introduction, and it's a pleasure to see so many familiar faces in the audience. And it's great to be back here in beautiful, sunny Stockholm. It feels tropical compared to Umeå. But I'll just jump right into it. So just to quickly introduce myself, I'm a bioinformatician, and my research group is interested in how the interplay between humans, animals, and our environment contributes to the spread of bacterial pathogens. So basically, we try to go from trillions of A's, C's, G's, and T's, so text data, and we try to use that to provide insight into the biology of bacterial pathogens. And if there isn't a method out there to do what we want, we basically just try to make it ourselves. So I'm super lucky. I get to work on a ton of really interesting microbiological problems. But today I wanted to talk about some super recent work my research group has been doing related to microbial secondary metabolite discovery. So secondary metabolites are compounds that microbes produce that aren't directly involved in microbial growth, development, or reproduction. But these compounds play really important roles because they allow the microbes that produce them to interact with each other, as well as with their environment more generally. And if this is a little vague or confusing, I always hate this definition, don't be worried because I guarantee you have definitely heard of at least one secondary metabolite before. Colobactin, for example, is a super hot topic in the gut microbiome world because it's been linked to colorectal cancer. Similarly, tilivalin, which is another microbial secondary metabolite, that's been linked to colitis. And then also my personal favorite, cerulide, is a toxin produced by some bacillus strains, and it's a common cause of foodborne illness throughout the world. If you've never heard of it before, maybe you've actually been sickened by it. It's one of the most common foodborne illness-causing agents in Europe. Or worse, maybe you've actually seen it on TikTok or Instagram, because apparently it just randomly blew up a couple months ago. And the only reason I know that is because I have a little sister who is way cooler than me. But not all secondary metabolites are bad guys. Many of the essential antimicrobial, anti-cancer, antifungal, anti-diabetic drugs that we rely on every day have been derived from microbial secondary metabolites. And these are just a couple examples. So whether they're really good or really bad for human health, we have a pretty big incentive to find novel microbial secondary metabolites. And as it turns out, the enzymatic machinery responsible for the production of many microbial secondary metabolites is encoded by physically clustered groups of genes within microbial genomes called biosynthetic gene clusters, or BGCs. So if you can kind of like see where I'm going with this, there's a huge incentive to mine microbial genomes and metagenomes for novel BGCs, because these novel BGCs might be responsible for the production of a novel secondary metabolite, which might have a really important role in human health or some really cool novel application in industry or medicine. So to that end, during my postdoc, which is increasingly long ago, I developed a tool called gene cluster prediction with conditional random fields, also known as GECKO. So I don't have time to go into the details. If you want to know more, you can just come and ask me. But basically, GECKO works by taking a prokaryotic genome or metagenome assembled genome as input. It identifies genes and protein domains within that genome, and it does this using a set of custom profile hidden Markov models that I trained specifically for this task. And then it identifies regions of that genome or metagenome that have a high probability of being a BGC. And it does this using a type of white box machine learning model called a conditional random field, or CRF. And then as like a final step, I just trained a separate random forest classifier to assign any predicted BGCs to one or more of these major biosynthetic classes. And I don't have time to show the evaluation metrics, also because I want to talk about new things today. But GECKO is more accurate and nearly an order of magnitude faster than previous state-of-the-art machine learning-based BGC detection tools. It's also more interpretable because it's a white box model, and as we show in our paper, that it's capable of outperforming the deep learning tools for this task, which is pretty cool. So when I started as a DDLS fellow at the very end of 2022, I had my cute little BGC discovery method. But now I somehow managed to convince like 10 people that I have some good ideas. So now I have a research group. And several of them have spent the past year developing some really cool new methods and tools that can help experimentalists discover novel BGCs. And just some cool new functional annotation methods more generally. So I figured I'd discuss a couple of these today. Note that all the work I'm going to show you is actually super duper new. It's not even in preprint form yet, but I'm sick of talking about old things. So I figured I'd like bring some new data and new problems that my group has been working on. But I'll start with some of our large-scale BGC mining projects. So back in 2022, when I started as a fellow, I had already mined over 1 million genomes and metagenome assembled genomes, or MAGs, for BGCs, including over 300,000 human gut genomes and MAGs, 34,000 ocean MAGs in this cool collaboration with researchers at ETH Zurich, and then roughly 1 million isolate genomes from the Progenomes 3 database. So this was basically every publicly available assembled prokaryotic isolate genome that was available at the time of this data freeze. However, now that I have my own group, I can do whatever I want, and they have to do all my crazy ideas. So with all of my newfound freedom, I was like, why stop there? Let's just go for it. Let's just mine literally every prokaryotic genome ever sequenced, just because we can, but also because we're guaranteed to find something new. Like some novel BGC that does something really cool and interesting has to be there. So that is literally what my PhD student Josephine has been doing for the past year, with some help from Adrian, who's a postdoc in my group, who's been really integral in helping us deal with all this data. So since last year, we've expanded our BGC dataset to include the entirety of the MOTUS database, which contains like 600,000 MAGs and isolate genomes. Plus, in collaboration with Zam Iqbal at the EMBL EBI, we mined his entire all the bacteria dataset, which contains nearly 2 million recently assembled prokaryotic genomes. And Josephine actually just finished the all the bacteria dataset like 2 weeks ago. We're still running some sanity checks on that before we make the data public. But so far, everything's looking good, and that's pretty exciting. So now we have predicted BGCs for basically every bacterial genome and almost every MAG ever sequenced and in existence at the time of this data freeze. So currently we're working on turning this into a BGC atlas so that other people can download and reuse and repurpose this data for their own experiments. But now that we have all of this BGC mining done, we kind of have a different problem. How do we make sense of all of this data? And that leads me to the next part of my talk. How can we help biologists and natural product chemists find their needle in a haystack BGC? So one that might be their novel antibiotic or their novel anti-cancer drug, maybe a novel toxin or a novel carcinogen. So to explain this problem, I just made some slides to visualize it. So let's pretend we have millions of predicted BGCs, which we actually literally do have. And these are represented by these gray arrows here. And let's pretend that this pink BGC here is some totally novel, never before seen BGC that equips some bacterium with the ability to produce some type of novel antibiotic. So we want to find this little pink guy here out of millions of BGCs at our disposal, some of which might be entirely new BGCs, unlike anything we've ever seen before, but a lot of them will be redundant BGCs. A lot of them will be things that experimentalists have already discovered and characterized and validated. So how do we do this? How do we find novelty in a virtually infinite pool of biosynthetic gene clusters? Well, one way of tackling this problem is to compare our unknown BGCs to a set of known BGCs, perhaps some experimentally validated BGCs which produce some sort of known secondary metabolite. And we can then use some sort of clustering algorithm to cluster our set of unknown BGCs along with these known BGCs into what we call gene cluster families, or GCFs. So by doing this, we can assign predicted functions to these unknown BGCs. And the rationale here is that if you belong to the same gene cluster family, or GCF, you're likely to produce the same or a highly similar secondary metabolite. So here, BGCs that fall within these colored clusters, they're experimentally validated BGCs that we predict produce the same or a highly similar secondary metabolite. And then using an unsupervised approach like this, we can also identify putative novel BGCs by looking for gene cluster families that don't contain any known experimentally validated BGCs. So in this example, we have like a cute little novel gray gene cluster family and a pink gene cluster family, which don't contain any experimentally validated BGCs. So in this case, we might want to take a closer look at these and then use some sort of additional methods or information to narrow down on our BGC of interest and predict its function. And this gene cluster family delineation approach, this idea, it's not really new. There's actually a couple tools out there that can do this, but they all have some major drawbacks if you're trying to use them for large-scale BGC mining efforts like we are. The first drawback is that they are ridiculously slow, to the point where you can't even scale them to millions of BGCs in a reasonable time frame, or sometimes it's even impossible to do this because of memory requirements. Also, many of these methods are not flexible. They're designed to work with the output of one specific BGC detection tool, which is anti-smash, which has kind of been the workhorse for BGC detection for, I think, it came out like 15 years ago. So if you're using these newer machine learning methods like mine or some other ones that are out there, you can't use these tools to identify gene cluster families because they're designed to work specifically with anti-smash output. I spent like years trying to make our tool, Gecko, compatible with this, and there's just so much, like 15 years of code that I really couldn't get it to work. So basically, it's not usable. And then finally, many gene cluster family delineation methods, they're just not designed with large-scale users in mind. They output really nice, cute HTML reports that are interactive, you can click around, but you can't easily access the raw BGC to gene cluster family mappings, which, like, as a bioinformatician, I need if I want to find novel biosynthetic gene clusters. So to overcome these limitations, I started this project during my postdoc with a really awesome PhD student, Martin, at EMBL, and we were like, let's develop our own BGC clustering tool, which we called High Throughput Delineation of Gene Cluster Families, or HTGCF. If you have a better idea for names, please let me know. We hate it too. We just couldn't come up with anything better, but we're taking suggestions. But this is the overall workflow. I don't have time to go into it for this talk, but if you're interested, you can come talk to me, or go talk to Josephine, my student, because she is actually presenting a poster on this. It's actually really, really quite simple at its core, but it seems to work really well, and I know this because I made Josephine spend the past year benchmarking this. So in our first benchmark, we wanted to assess the accuracy of our clustering tool, HTGCF, relative to Bigscape, which is a BGC clustering tool optimized for accuracy. And we evaluated these two tools on this manually curated dataset, which Bigscape published in their original paper back in 2020. And here we can see how our tool, HTGCF, clusters these BGCs into manually curated gene cluster families, so like ground truth gene cluster families. So basically, the more light shading you see along this diagonal, the better. And as you can see, we do really well, and this is actually on par with what Bigscape, the state-of-the-art, does on this same data set. But then in a second benchmark, we wanted to assess the speed and memory usage of our tool relative to Bigscape, which is that state-of-the-art BGC clustering tool that's optimized for accuracy. But we also tested a less accurate tool that's optimized for speed, which is Big Slice. And we did this by clustering a couple thousands of BGCs from a bunch of different prokaryotic genera. And here you can see how our tool, HTGCF in pink, if you can maybe see it, the bars are really small because it's really fast and really good. But you can see how HTGCF compares to Bigscape, which is the blue bars, the tool that's optimized for accuracy, and Big Slice, which is less accurate but faster than Bigscape, and that's in green. And as you can see, we're roughly, like 47 times as fast as Bigscape, but also more than 10 times as fast compared to Big Slice, which is the method optimized for speed. So this is like really promising. But great, we have comparable accuracy to the state of the art, and we're over 10 times faster. Let's see what we can actually do on a large data set. So we used HTGCF to cluster over 2.8 million BGCs detected in roughly 1 million prokaryotic genomes from the Progenomes 3 database. So this was basically every assembled prokaryotic genome that was available at the time, just to see if we could. And we did. So this is a UMAP, which shows the gene cluster family representatives identified by our tool in nearly 3 million gecko BGCs from 1 million prokaryotic genomes. And I didn't really show it in this picture, but the vast majority of these gene cluster families, they don't contain any experimentally validated or known BGCs, meaning that the vast majority of this biosynthetic landscape has just never even been touched by experimentalists. So our new method can scale to massive data sets, which is pretty cool. And then another great thing is that you don't even need to use biosynthetic gene clusters as your input like we do. You can actually cluster any type of genomic segment. So just for fun, I made poor Josephine cluster all secretion systems in this Pasteur database. And here again, we show it in 2D space with a UMAP. But jumping off of that, because Martin, my PhD student at EMBL, and we spent years developing Gecko and trying to make the code really user-friendly and flexible. So now we actually have a really nice piece of software, which we could hypothetically use for other genomic sequence segmentation tasks. So I'm really interested in seeing if we can retrain Gecko, the underlying conditional random field, for other types of annotation or genomic sequence segmentation problems. So we've actually been doing this in my group. For example, Florian, who's a master's student and soon to be PhD student in the group, he actually retrained Gecko to find some novel exotoxin encoding genes in bacteria. This is super duper preliminary. We just used his model to mine the MOTUS database, so all these isolate genomes and MAGs, 600,000 of them, for exotoxins. And we haven't looked at any of the results yet, but I'm really hopeful we'll find something cool. And then finally, really recently, just for fun, I was bored. So I retrained Gecko to find novel secretion systems. But again, we just finished mining the MOTUS database with my model, and I haven't really looked at the results yet. But you can hypothetically retrain it for other types of tasks. I think prophage would be a next logical jump. This is what I do when I'm bored and watching Real Housewives. But wrapping it all up, with fast and scalable BGC detection methods, we now have unprecedented amounts of BGC data, which is super duper exciting because there's definitely novelty there, but it also requires extra tools and extra methods to make sense of it. And as we venture into this new era of BGC research, it's really easy to see that the vast majority of the microbial biosynthetic landscape has just never been explored, meaning that there's potentially tons of novel microbial secondary metabolites out there for us to discover. And then finally, sequence segmentation models, including white box models like conditional random fields, they can be used for genome annotation tasks beyond biosynthetic gene cluster detection. And this is something I'm really interested in exploring with my group in the future. So as I wrap things up, I'd like to give a special thanks to my collaborators, especially Martin. He's an absolutely amazing bioinformatician who was integral to multiple projects that I discussed here, and he could have given this talk and done a better job. Also Georg, who came up with the original idea for Gecko as a BGC detection tool. All of my funding agencies, it's nice to have money, thanks for paying me. And a special final thanks to my amazing group. A lot of you are in the audience today. None of this work would have been possible without them. Josephine did a lot of the work that I presented here today, but all of them have been integral in helping out with these projects, but also their own projects, which I sadly didn't get a chance to talk about. But come check the posters, because they all have, or most of them have, are here and have some. And thanks to all of you for listening. Sorry.
So thank you, Laura, for a very interesting talk. Happy to hear there's lots to be discovered within the intriguing world of microbiota. So any questions from the audience now that you're refreshed with coffee? We have one over here.
Thank you for the talk. I'm a data scientist, so I'm minority here. So you mentioned a lot about clustering. So I'm interested to know what kind of clustering method are you using? And then why do you choose that particular method?
Yes, so it's so simple. So basically there's a couple sequence similarity based deduplication steps that we run using MMSeqs, if you've ever heard of that tool. It's specific to working with sequences. But the final clustering step is basically just hierarchical clustering. We tested a bunch of other methods, and this one worked the best by far. We were kind of shocked. So it was really cool. You didn't have to use very fancy things to get really good results, and it was fast, and it seemed to scale well when you implement these deduplication steps. So it's actually like the simplest pipeline ever, and beat the state of the art. So we're like, okay, let's stick with this. But yeah, very, very simple hierarchical clustering with some other bioinformatics steps stacked on top.
Any other questions at this point? Okay, please go ahead. There's one.
Any ideas how to start testing them in a lab at the large scale?
Oh my God. That's the hardest part. Honestly, like you can find the coolest things ever, but it costs time, it costs money, it costs hands in the lab to test these. And I'm not an experimentalist. It's so far above my head. So I have a couple collaborators, but I don't have a good solution. Basically, right now, what we're doing is using these methods to narrow in on some possible candidates and try to, find things that are novel enough that they're like, this is exciting, but not so novel that they're like, this is scary and I don't want to waste my time and money testing it. So I would say that is actually a huge bottleneck, is the experimental approaches, because often what you have to do is basically clone it into E. coli or your organism of choice, try to get this thing expressed, and then get a metabolite out of it, and that is not trivial at all. So that is actually, I would argue, the harder part. I have no good ideas for that.
All right. There is one final question, and then we need to move on.
Hi, great presentation. Now that you have all these BGCs, have you thought of creating a database so that we can directly map all metagenomic reads to all of these BGCs, especially to look at which BGCs are higher or lower in specific individuals or cohorts?
Exactly. That is actually something. So we're making it a massive atlas. That's the goal. It probably will be quite some time to figure that out and finish that. But I really think that's a really cool idea. And that's something I've played around with, looking for BGCs that are enriched in certain disease states. And absolutely, that's something we could consider adding, for sure. I looked at BGCs enriched in like colorectal cancer and type 2 diabetes. I never published it because I couldn't get any experimentalists to test anything. But that's a great idea, and definitely something we would consider doing.
All right. With that, thank you, Laura.
Thanks so much.
And next, I'm happy to introduce Jakob Vogel, who is also a DDLS fellow at the Lund University, and he will be presenting on the topic of leveraging clinical data towards biological insights and precision medicine in Alzheimer's disease. Most welcome. Looking forward.
Thank you very much. Thank you for the introduction. It's a real privilege to be here with all my DDLS colleagues and peers and friends. I've been a huge fan of watching your amazing presentations and seeing the cool work that you've been doing, and so I'm happy with this opportunity to sort of familiarize you with what I've been doing. And sort of like Laura, I'm going to be focusing almost exclusively on work that we've been doing since I've arrived at the DDLS program less than 2 years ago, so kind of unpublished new stuff. And so the focus of our group is really on neurodegenerative diseases, but most proximately Alzheimer's disease. And just this is a generic slide, but I think it can't be understated how big of an issue this is, right? So 1 in 9 people who are older than 65 are living with Alzheimer's disease, 1 in 9. And a quarter of the world's population will be above 65 by 2050. So you do the math. I say that partially because I couldn't be bothered, but also because I think it's pretty obvious that this is not just an individual tragedy of a disease, which it is, but it's really threatening to become a pretty enormous social and economic sort of catastrophe. And so because of that, there's a lot of people that are working to stop it all around the world. Just like with any other sort of realm of biology, there's people who are using cell models and animal models, and of course there's people who are using really clinical data that's been collected from actual patients. And really by design, my group is mostly focusing on this latter category. And I think we're all sort of aware of the disadvantages of that, right? The ability to do causal experiments in this setting is pretty limited. You sort of have to get a lot of individuals together and observe sort of natural experiments and produce hypotheses based on that. It's a lot more challenging. And of course, our resolution when asking these questions is pretty limited. We can't dig about as deep as we really want to when we ask these questions. That's why we, of course, have these cell and animal models. But I think the advantages of using clinical data is actually quite a bit understated. First of all, the armamentarium of tools that we can use to probe human physiology is growing, and it's actually at a really rapid pace. And I think there's a lot that we can actually explore to really generate hypotheses using clinical data. And I think that's very important because of this first point here, this ecological validity. It's really useful to have these cell and animal models of Alzheimer's disease, but none of them are even close to perfect. They just don't capture the complexity of the disease. So we think that by using our data science, domain knowledge combined with computational and statistical skills, we can sort of tease out new information about these diseases. We're also more proximal to sort of these clinical tools, so we can bring them to the clinic faster. But we also want to generate new hypotheses from the people who actually have this disease and sort of feed that back down to people who can sort of validate it in models. So that's really what my lab does. We take clinical data from neurodegenerative disease patients, and on the one hand, we actually try to learn new things about the disease and formulate new hypotheses, and we also try to build clinical tools as well that we can sort of feed back to people running clinical trials or help with diagnosis and prognosis and so forth, and sort of contribute to this precision medicine thing that's going on. So we do that in a lot of different ways, and I'm going to talk about some of these approaches that we use. This is by no means comprehensive. We're doing a lot of different things, some of which are related, some of which really aren't. But this is some of the main foci of the group, and I'll hit on a couple of these with the experiments I talk about in the talk. But first, I think it behooves me to just give you a really, really quick crash course on how Alzheimer's disease works, so the stuff that I present makes a little bit of sense. So on the x-axis here, you just have the time course of Alzheimer's disease. So from preclinical, where there are no symptoms, but the disease is sort of burgeoning. Prodromal, when you have some limited symptoms, and frank dementia, where you have strong impairment and function, inability to function, basically. And on the y-axis, you have changes, abnormal changes in different pathologies. There's amyloid beta plaques, which build up very early in the disease process before you have symptoms. There's tau neurofibrillary tangles. These build up a little bit later and are much more proximal to neurodegeneration or atrophy. This is the loss of brain cells and cell population, and it's this loss of neurons that leads to cognitive impairment. And of course, there's all these other things going on in the background, right? Neuroinflammation, neurovascular dysfunction, all these other things where we don't actually know if they are quintessential parts of the disease process or if they're just sort of epiphenomena that occur in the background because an organ is being damaged, and a lot of people are trying to figure that out. But importantly, for our purposes, all of these factors can now be measured in living human people with biomarkers, right? We can use neuroimaging like PET and MRI to look at so the presence and the distribution in the brain of this amyloid and tau pathology. MRI we can use to look at sort of neurodegeneration, and we can use proteomics to start to get a little bit at some of these ancillary things that are happening in the disease as well. And so that's what the lab tries to do. We try to use this to learn more information about the disease. But interestingly enough, even though we have these 3 hallmark pathologies and we know them very well, we still don't really know that much about how they interact, how they lead to one another, and why they lead to cognitive decline. And so what we are trying to do now is build what you can think of as like in silico models, right? If we can't do experiments on humans for obvious ethical reasons, what can we sort of create an environment that recapitulates the primary disease processes? And we do that by looking into the literature and taking the things that we hypothesize to be true or that we know to be true about the actual disease pathophysiology and see if we can build that into some sort of model. And so, for example, we think that we know that tau pathology spreads through the brain using the brain's own neuronal architecture, right? So it actually spreads down axons to the synapse, crosses the synapse to the synaptic partner, and that's how it's actually moving through the brain. And we can model this spread because we can use neuroimaging to actually model human brain connectivity. We can figure out anatomically which brain regions are communicating, and we can model the spread of pathology through these communicating neurons. And so we know that these amyloid tau and neurodegeneration interact somehow. We have some hypotheses of how they interact, and we're sort of building this disease simulation model that's actually fairly parsimonious, fairly simplistic, that just involves amyloid potentiating tau and tau sort of driving neurodegeneration, and a little bit of this axonal spreading there to describe the transport. And there's sort of a real focus here also on identifiability of these parameters. And so what's really interesting is that if we sort of bring this simulation to the brain, and we sort of start in this initial position, and we allow the simulation to take sort of to happen. I'll show you what happens. On the top you have amyloid, in the middle you've got tau, and on the bottom you have neurodegeneration. And so first amyloid builds up very early on, just like in the real disease. Then we start to see tau in focal areas. It spreads to certain parts of the brain, and then is followed slowly thereafter by neurodegeneration, actually rather quickly by neurodegeneration. And what's really cool about this is that the regions that become that get pathology are actually emergent. We don't tell this model how much each region should get of pathology each region should get. This sort of emerges from this interaction between our sort of parameters and this connectome-based spread. And what's interesting is some of that's based on experiments that have been done that we're integrating into this model, and some of it is based on just tweaking things and figuring out, well, what if we do this? Does that actually fit the data better? What if we use this sort of hypothesis of how tau influences neurodegeneration? And so, what's really cool about this is when we fit it to real data, real patient data, people that have these biomarkers over time longitudinally, it fits very well. So each one of these dots is a brain region. The x-axis is the actual change in pathology in that brain region, and the y-axis is the predicted change in pathology by this model. And you can see we're actually doing a very good job predicting how much pathology is going to build up at a regional level in the brain, which you can imagine there's a lot of different sort of clinical use cases that we can apply this to, from clinical trials to prognosis and so forth. All right, so moving back to our sort of model of Alzheimer's disease, I mentioned that we can use MRI to measure sort of changes in neurodegeneration. So MRI, you're probably very familiar with it. Brain MRI is used all over the place in clinics. If you come in complaining of some sort of cognitive impairment, it's common you get a CT or MRI to rule out things like stroke or hydrocephalus. It's actually used in treatment planning for different disorders, and researchers use it to basically image macro-scale neurodegeneration of different regions and cell populations. But so if you look at a normal 3T scan, right? So this is a 3 Tesla scan. We're looking sort of from the top down onto someone's brain, right in the kind of middle of the brain. And 3 Tesla is the magnet strength, and that sort of gives us information about the resolution of the image. And if you know your neuroanatomy, you see it's not bad. You can see some cortical structures here, the thalamus, the striatum. You can see the cortical ribbon sort of going along the edges, but it's kind of blurry. And so we also have this sort of 7 Tesla MRIs. You never find find these in the clinic, but they are, as you can see, a lot sharper. They have better resolution, but they're a little bit unwieldy. They're difficult to use. They're expensive, and maybe it's not that obvious to perceive the difference here. But if we look at a structure like the cerebellum, you can see the 7T. There's this really nice sort of branching of this structure that's just not even really visible in the 3T. So we started wondering, it would be great if all of these hospitals and research centers that have 3T scanners could get to 7T resolution somehow. So we've been messing around with using different deep architectures and paired sort of 7T and 3T images to try to sort of synthesize higher resolution images. And it's been fairly successful so far. We were able to actually, this is a synthetic image based on this 3T MRI. It was not involved at all in training. And you can see there's an enhancement in the contrast, the sharpness of the image. You can even see some of this sort of branching in the cerebellar structures. So it's pretty cool qualitatively. We actually asked a bunch of neurologists and radiologists and MRI scientists to sort of tell us what they think, right? Rate these images on how good they look and how detailed they are. Unsurprisingly, they said the 7T, the higher resolution images, were better than 3T, the low resolution images. But what surprised us a lot is they actually consistently, especially for our GAN model, rated our synthetic images as even better than the 7T images somehow. And we think that the reason is because there are all these artifacts from the magnet caused by the 7T, and those are not there in the synthetic images. And these models were also doing better compared to some of the models that were out there in the literature. So that was pretty exciting. But that's all very qualitative, right? What about quantitative sort of proof that this is somehow useful or interesting? So often we segment the brain into different brain regions because we want to understand what they're doing and how they're involved in diseases. And we use automated segmentation because to segment just one brain region for one person takes 15 minutes to an hour, and it's just not feasible. So we use these automated segmentations. We did happen to have about 12 brain regions manually segmented, and we said if we basically do an automatic segmentation of our synthetic images and the real images, which one gets closer to the sort of ground truth of the manual segmentations. And this is a busy slide, so I'll just focus on the sort of true 3T versus our synthesized model with the GAN. And you can see in 9 of 11 cases, the automated segmentation made a segmentation that was closer to the ground truth. So this is some quantitative evidence that this might actually be useful beyond it just looking nice. All right, so I'm going to end the talk with sort of focusing on one last aspect of the sort of Alzheimer's disease pathophysiological process, which is these ancillary measures, which we think we can sort of track a little bit with proteomics. So we do a lot of proteomics in the lab. This is an example of cerebral spinal fluid proteomics. In Alzheimer's disease, we characterized a bunch of proteins involved in the very early stages of Alzheimer's, where you just have amyloid in the brain, sort of asymptomatic, and then the later stages where you have both amyloid and tau. And we characterized, which ones are involved in these different processes, including 15 core proteins that were continuously upregulated as or more abundant as the disease progressed. And so we did a lot of characterization of these proteins. I'm not going to focus on all this stuff. I just want to zoom in on one little area. Here we are looking at the sort of amount of the protein in the CSF, the abundance in the CSF, as compared to how much pathology is in the brain. And we were interested in this one protein, SMOK1, that was very associated with A-beta pathology, this very early pathology of Alzheimer's disease, but not at all with tau, the sort of later part. And we thought, this is a pretty unknown protein. We didn't see this in any of the animal literature or the cell models, but it's associated with a very, very early part of the disease, and it's very salient. So we wanted to dig into it more. We took a single nucleus RNA data set, Alzheimer's data set that was published, and to figure out what cells are actually expressing it. And it turned out that there's a type of brain cell called oligodendrocyte precursor cells that no one really pays attention to in Alzheimer's disease that expresses this. Not only does it express the transcripts for these proteins, but it does so more in response to Alzheimer's disease pathology. So we basically use this transcriptomics data set to replicate what we found in this sort of living human proteomics. And we went on to, with collaborators, do some immunohistochemistry and found indeed Alzheimer's patients were expressing this protein more around the OPCs, and it was actually even co-localizing with the amyloid plaques in these people, suggesting an actual interaction going on. And so we went from this like unknown or mostly unknown protein that we didn't know was interesting in Alzheimer's disease to learning sort of what it's doing and what cells are sort of expressing it, and we're going to continue to study it because we think it's important. That's just an example of how you can go from this bioinformatics to actually learning something new. And finally, all of you will know a data scientist is really only as good as the data at the end of the day. And so we've been very fortunate to be working with this Gates Ventures Global Neurodegeneration Proteomics Consortium data set, which has about 40,000 blood samples from people with different neurodegenerative diseases from several cohorts. And it's going to be released to everyone in 2025, but we're fortunate to have some early access to this data set. And it's really the perfect setting to design a multi-disease classification, right? You've got these different neurodegenerative diseases. They're hard to diagnose. Differential diagnosis is very difficult. What if you can just take a blood test and with a couple of proteins be able to say, hey, we think that there's a strong probability that you've got this disease and not this one, or maybe you have both. And so we're using these sort of deep multi-output classification models. We can use the embeddings of the last layer to help us with some transfer learning that we want to do, and we can do some latent space modeling. And we're sort of very early on in this process right now, but we're already getting quite good sort of multi-class diagnostic accuracy. And we're finding that this approach really generalizes out of sample and to completely new sites better than your sort of out-of-the-box machine learning approaches, which is good. And we can even sort of try to understand what's going on with the embeddings, which I'm projecting here in 2 dimensions. You can see that there's these sort of clusters based on disease. Alzheimer's disease on the left, Parkinson's on the top, controls on the right, and FTD in the center. And what's cool about this is, we're trying to figure out these dimensions are meaningful, but also, what about these false positives? Maybe this is actually a failed clinical diagnosis. The labels are actually wrong in these cases, we think, and maybe the proteomics are giving us a little bit more insight to what's going on under the hood in these patients than the clinical diagnoses are. So we're investigating that as well. So I'll wrap up and just say, this is a small sampling of some of the stuff we do in the group, and there's a lot of other stuff that we're interested in, but if you guys are interested in it, I'd love to talk to you. You all do such cool stuff, and I'd love to collaborate and see what we can do together. And I'd just like to thank, first of all, the group, the lab. They've been a bunch of really incredible people, some of whom are here, that have been really driving this work. And of course, the funders for, as Laura said, paying us. So thanks so much for your attention.
So thank you, Jakob. Very impressive and interesting. And I already see a question over there.
So there is this increasing trend of unproportionally increasing the enrichment of diseases within the years, right? So you showed, right, that by 2050 we're going to have a quarter of population that is having some sort of dementia.
Well, a quarter of the population will be old. And so when you're old, you are much more likely to have these diseases. So 1 in 9 people who are old have Alzheimer's disease, and a quarter of the population will be old by 2050, basically.
Okay. So, but you think that the disease is not kind of, well, 2 questions. Whether these diseases the situation is getting worse, like in terms of proportionally to the old population. And the second, why is that in terms of like, I'm pretty sure you can point out some factors, right, that leads to that.
So it's actually not getting worse. It's getting a little bit better as we're, getting more. For example, there's a strong component of neurovasculature in Alzheimer's disease, and as we've gotten more heart medications going on, we've actually seen decreased incidence. But the problem is that the world's population is growing, and there's going to be just so many old people in the world, many more old people than there are today. And even if the incidence is sort of going down, there's still going to be so many cases to be dealing with, and it's a big problem. But in some lower and middle income countries, actually things aren't looking better. I would say they're looking worse. And what are the factors of that? We know that there are some modifiable risk factors. I mentioned sort of heart disease, and there's other medical ones like diabetes. There's a lot of stuff related to pretty much the same stuff that's related to everything else, diet, exercise, and so forth, that are modifiable. And a lot of people think that there's a lot to do with sort of global pollution that might be contributing to some of these diseases as well. But actually, the problem is just the amount of people that will have it. That's the real problem.
We have another question over here.
So hey, Jake, that was really fantastic. First of all, I fully agree with the kind of a emphasis on human data as much as you can, and as beautifully you showed. But in a sense, you looked at all of these things in the context of biomarkers and the diagnosis of disease. So if you just took proteomics on the sort of a individual protein level and you looked at the images and all the features in the images, can you predict certain links between the two?
Yeah, it's on the list of things to do. But there's a degree to which the resolution combined with the nature of how these pathologies distribute, there's something a little bit stereotypical about it, right? So the more of a pathology you have, the more predictable it is where that pathology will be. And so what's actually very difficult is disentangling where the pathology will be, which is actually quite relevant to clinical presentation versus just how much you have. And so a lot of these proteins so far have just been associated with the amount of pathology you have, which is more or less how fast you're accumulating it. Some of them we're sort of digging into why does it accumulate more in this part of the brain and cause these symptoms versus others. But yeah, we're something that we'd love to do is just do something very data-driven, like you're talking about. Take the proteins in one data set, take the brain data on another, and just throw it together. And I will say the explainability of the CSF protein data for biological phenomena is beyond any other marker that I've looked at before in terms of it being able to explain what's going on. So it's super interesting because it is high dimensional, but it's also pretty high rank, I would say. So.
And we have a final question over there.
Thanks, Joel. Very interesting talk. So first question goes to about when you increase them from 3T to 7T, did you experience any artifacts? And the second question is, could you give me a little bit details on how you produce synthetic data or simply convert the two sets to produce that?
Yeah, so yes, the first question about the artifacts, sort of in a way, so the 7T images, you can actually see blood vessels, and they're very hyper intense. And so the model basically figured out, well, synthetic 7T should have blood vessels, but they just kind of put hyper intense specks all over the place. And so we actually ended up just thresholding the images, which got rid of those because it was just kind of hallucinating these hyper intense blood vessels, which we don't really pay attention to anyway. We've also seen at least one example where the model confused a just regular heterotopia, basically some gray matter inside the white matter, as a lesion. When it wasn't actually a lesion. So there is some risk of kind of hallucination, but not a ton so far. And what we've seen more of is there's tons of magnetic artifacts that really ruin the images that just don't really exist because the 3Ts don't have them. So we've seen that. In terms of how we're generating these models, we've used a bunch of different architectures. We use UNets, we're using GANs. There's certain aspects of the architectures that in terms of like attention masks and sort of like certain layers that we've added that have been really helpful and some that have done basically nothing. We've probably spent too long tweaking some of these things and most of them don't change anything, and then one day you just kind of do the right parameter and all of a sudden everything changes and we don't always know why. So yeah, but I mean, if you want some like really nitty-gritty details, we should just chat about it after this, and I can show you the architecture.
But thank you. I know there are more questions in the audience, but please do save them for the coffee break. And in the interest of time, thank you so much for this talk. So I'm happy to welcome Alexei Selzniak, an associate professor from Chalmers and a former SciLife Lab fellow, to the podium. And he will be talking on leveraging generative AI for applications in synthetic biology. Looking forward to your talk.
Hello, thank you. Thanks a lot for this opportunity to present our work. It's actually 13th of November. I did, yesterday evening this presentation and just looked at the date yesterday. So today I'm gonna little bit talk what we do. So generally, kind of when you look at the synthetic biology problems and protein engineering problems, what you typically want, you want to optimize the DNA or protein sequence. And normally how you do it in the lab, you take some parent molecule and basically diversify it, and through many iterations, you eventually come up with a sequence that you put to production that basically fits to your desired objective criteria. Unfortunately, kind of this is called traditional engineering, but it actually resembles something what like in the gold rush when you back in the days you had this basically plates where you were doing gold paintings, so to fish out some particles of gold. So, but not rather engineering. So what I believe what is possible to do with as today was presented several times, how can we actually do a digital twin in the computer and I believe that AI can facilitate that for sequence design specifically to kind of to make AI type of sequence engineering that is done in silico, which is more efficient and easier to do than if you would do that just only in the lab. So basically, typically what we actually working on two problems, and normally in synthetic biology and organism engineering, you do very often do two things. You basically put DNA into the cells, and you in order to express proteins. This is in fact probably most popular experiment in molecular biology. You basically take the DNA and you put it to the cell in order to express or to secrete protein or to locate it to your designated organelle. And the second thing, you also want to improve those proteins. One could be making them more stable or more active, et cetera, et cetera. So first I'll talk about a little bit what we do in an expression level. And if you look at the kind of what is AI, how AI are doing the faces nowadays, right? We know that they are all beautiful and so on. Unfortunately, with the DNA, we still kind of stuck things together quite randomly, even though it's a beautiful Picasso picture, but probably, cell recognizes that this is some sort of not real thing, and that eventually it doesn't work that well. So basically, to deal with this problem, we look at the gene expression data, and I want to pay attention to this figure. So I would really want to basically, if you look at this kind of the bumps, right, what they represent gene expression level variation across this number of RNA-seq experiment, and they arrange basically by average expression. And you can see some genes, so this is just represent one gene. Some genes are low expressed always, and some genes are very high expressed. And you see that the low gene expression will never be here, right? It will just stay always there. And the difference between low gene how low gene expressed and high gene expressed is 7 orders of magnitude, which is huge. And the same, like, if you look at the all experimental data available, for instance, in yeast, you look like kind of in this fold chain. So experimental variation will be 2/3 of them just within the fold change, basically, and the ratio between the lowest expressed genes and the highest expressed genes will be orders of magnitude. And the idea, we can just read the DNA and predict the gene expression levels just on average how they are. And the idea, and indeed you can actually, and that's show the gene expression data. So each of these dots is a gene that is expressed in a cell, and just by reading the DNA, we can tell on average how much it is expressed, and this can be done for multiple organisms. And what you can do with that, you can basically then look at the neural networks, try to figure out what they learned and extract motifs, patterns that are informative about gene expression. And then you want to kind of figure out, is there any logic how the language of the DNA is organized, and you can then try to do these simple statistics that usually used for market basket analysis. So usually you want to find associations of items that are more frequently together bought than any of them on alone to create all sorts of marketing strategies. And often what happens when young fathers are going to buy beer, they often buy, when you go to buy diapers, they often buy beer together. And basically, we turn those rules. You can identify those co-occurring basically motifs present in different parts of DNA that are more present in the groups than any of them on alone. You can see the parts that are highlighted, the terminator and promoter. There is a quite hard bar. So there are parts that are kind of in both section of that. It's a shame that the laser doesn't work. But basically, what it means that you can have this, the blue one highlighted in blue, it's a transcription factor binding site that depending where the motifs are and what are they, you can have a very large expression levels difference. So they thought, typically in molecular biology, you look like, okay, you have strong promoters or weak promoters, and you have kind of this belief that there is a strong promoter and so on. So we basically wanted to find if there is any interaction between those and designed So we kept native promoters as they termed either strong or weak, and we changed the terminators based on what we think, how can they make changes in the gene expression. Basically, to cut story short, we made the very low expressed genes very high expressed just by changing terminator. So it's not only a notion of like what is strong promoter or what it's just generally how the gene is organized. And when we start looking then together, kind of going deeper, I want always showing kind of this figure with the faces, and that's kind of, you can make some faces look younger, older, and that's kind of a little bit old slide, well, a couple of years old just. But in essence, we wanted to design DNA in order to have a specific levels of gene expression. Can we navigate the gene expression landscapes for that? And this is what's shown here. Basically, you have a t-SNE plot. Each of these dots is a sequence that you navigate towards your desired level of expression. So all of those dots, you start from blue, and then they went to the red ones. And then black ones we took and put them to the cell, express them, and measure the expression levels in the yeast. And basically, you see that those which are with the gen, basically those are generated. And you can see that some of them is extremely different DNA. The yeast doesn't have anything like that, but actually our designs are expressed more than ribosomal genes, and ribosomal genes are basically to make up to 1/3 of the proteome. So you can actually do quite control expression in this way for many all sorts of applications. So in general, use generative AI to jump for many years of evolution. So I'll talk a little bit about gene expression now. Now, what about the proteins, right? So typically, again, this protein optimization often depicted as climbing this landscape, complex landscape, mountain landscapes. But in reality, in the lab, what you actually do when you do this evolutionary engineering is actually introducing mutation and screening for dead proteins most of the time, because whenever you introduce mutation, often what happens with the protein, it kills basically activity with that. And the idea was, can we do a little bit better just by using generative AI the same way how we can navigate the phase space? Can we actually navigate the sequence space? And we did this already more than 5 years ago. So that's basically, this histogram shows so all those lines are experimentally designed sequences. So we build a generative adversarial network with some attention back then. So this was the first example how was attention introduced to GANs with sequence design. So and basically those with the numbers there, those are actually experimentally active ones that are working ones, and these are just some more examples that worked in the lab. And that was a preprint was published in 2019, so quite some years ago. And back then, we couldn't basically publish that anywhere. Bioinformaticians only knew something called hidden Markov models, and not really work much with generative models. And that was a bit. And now you can see that basically you have in every single week publication about some LLM generative model in the top tier journals regarding the proteins. You saw now with all the Nobel Prize victory. So that was definitely the interesting field to work with. And we basically our methods was a part of largest course on the internet for generative adversarial network, kind of to show example how you can do applications just besides other domains. So basically, okay, we can generate the design, right? The pools. We can moving from lab process to computer process. Can we now improve variants? So what we now working on is in unpublished data. So, but you heard probably what is some of you heard what is neural style transfer. So basically, you can it's a domain of computer algorithms where you take a image and represent it in style of other image. So for instance, here you can mix make a Mona Lisa in style of Van Gogh, or do something like that, which has many good applications. Also, you can imagine very bad applications. So what we want to do basically with that is to do protein properties transfer. In this case, is a transfer of the temperature transfer. So can we make actually take any protein that is mesophilic and make it thermophilic. And the challenge with that in actually in biology, so typically how you would do it, normally traditionally, you take it, sequences of your desired proteins, and you go back some time in evolution in order to find something that was for prehistoric, where the earth was hot. So then typically you can reconstruct the sequences using ancestral sequence reconstruction and basically make the enzyme, for instance, thermostable. And that's a very important property for industrial applications. The problem is that if you look at the entire UniProt, which is in blue, you have a less than 1% of annotated sequence. So you basically don't know actually anything about what is the sequence temperature. Even if you try to annotate it, the problem that if you go kind of looking at the sequences, you have basically in the entire UniProt you have very few sequences. So the blue line is one orthologous group represented, the fraction of orthologous group represented with having at least 2 sequences. So basically, with 2 sequences, you almost have none of the annotation. So I will not go to the details how we did it, but you can actually use some metagenome sequence data and labeled where where depending where it was taken from. For instance, some sequences are taken from volcanoes, right? And then if you take the metagenome sequences that are taken from volcanoes, you know that species are growing there at 100 degrees. So that's automated labeling. You can cleverly label data, and then you do some AI, which I will not go to the details, but in essence, what you can do, this is experimental data. What it shows that you can take it and do a style transfer. So the solid lines are wild-type sequences, and the dashed lines is the sequences that we designed, and we can actually do the temperature style transfer on a sequence for multiple examples. In essence, we want to create a general foundation models of temperature transfer. So now we are having a trouble of training it with available, even with a large computer that we have here in Sweden. The Berzelius is very busy with these tasks, and scaling is a problem. But in essence, what we want to show here also, you can kind of evolution. You can see on those. This is UMAP projection of embeddings of those, and you can see these are wild types where you can see there are patches that are only have very few examples, and then you can actually take those and basically expand it. And even though in our training data there were no any single thermophile, we can actually expand it, the space of the sequences to make the thermophiles. And so generally it is possible to improve variants using generative models. How about like what is about there is one though problem. In computer you can do many things, right? We can create millions of sequences in our virtual twin, right? The question, how do we make those sequences actually that are working in the lab? How to prioritize? And this is demonstrated in this figure, which would probably make sense, right? Like if a dog on the moon is fine, but it doesn't make sense because there is a moon on the background. So how can the dog sit on the moon and have a moon on the background? Which very hard problem to identify in proteins. We cannot look at the proteins and say that protein is good by eye because we don't know anything. Even one mutation makes it inactive and so on. And many people, what they do, they rely on AlphaFold as a kind of a golden standard. Okay, if it can make a structure, then maybe this is a functional protein. Everything is good with that. The problem with that, we designed the 500 make proteins, express them in the cell. You can see that identity doesn't really matter. You can have few mutations that kills the activity, or it doesn't express, or God knows what is happening there. Or you have, and all of them have very perfect AlphaFold structures, right? So it doesn't really matter. AlphaFold will produce always a functional, nicely looking enzyme, we respectively that it's actually in the lab, working in the lab or not. So we wanted to, this was recently published, so we wanted basically to find and to create a silico metric that would tell that things are working in the lab. So we could in the computer start screening things and prioritize it. So it's like having what was told today, having some kind of a scoring that would optimize your likelihood of things that are working. So we tested many things and came up with well, actually, language ESM models are working quite fine, much better than some alignments and some structural properties. So the way how it works, basically, you have a pool of your generated sequence. You can do some filtering based on identity. Then you can filter out using different filters that we tested. And in essence, with this pipeline, you can use it for in any protein, but it increases almost doubles, or sometimes many more times, the success rate that your designs will work in the lab. And we tested that in many sequences. So, and that's just to kind of what I want to show, that we can, I believe that we can actually make biology programmable by introducing AI into this process. And this is another example what I want to show, that things are actually working quite well for other applications. So we can actually design viable proteins. So this is a piece of plastic put to the cell, and then we engineer the enzyme that can degrade completely plastic coming from all different sources, including textiles. Within just a couple of days, you get basically a solution that is free of plastics with a monomer that were the actual plastics. So you can recover that monomer and use it for applications. And this is, of course, done with all these wonderful people who are sitting here in the lab. And I would like to also here in the audience, and I would like to thank funding sources and you for listening. Thank you so much.
Thank you for a very interesting talk, Alexei. Do we have questions in the audience? We have one over there. Karin is running with the mic.
Hi, thanks a lot for the talk. I was wondering, I didn't get the point of the plot you showed once, like native sequence recovery and then the PLDDT. What was on the y-axis?
So it's just a cumulative. It's a cumulative. It's like a distribution, but cumulative, right? It's a fraction of sequences that are active and fraction, yeah, depending on whether active or inactive. Yeah. So it's a cumulative distribution.
Thanks. I'm a bit surprised that there was no correlation between, for instance, PLDDT and some, experimental success rate. I'm not too much concerned about, PLDDT not correlating to the enzymatic activity, but to expressibility and solubility. Should work reasonably well. So I guess Kevin released a paper a couple of months ago, Protein Bench, where they were predicting structures with MSA. Did you do MSA mode, AlphaFold 2, or single sequence mode?
MSA.
Well, I think that's the reason.
Well, Kevin was an author on that paper.
Okay.
we can chat about it later. Because I don't think that's the. I guess they use the same data to benchmark it, right? So, but yeah.
We can talk about it later.
Yeah, that could work, but I'm just saying that it's not the best. AlphaFold works to some extent, but it's not the best way to predict what the sequence is active or not. That's what the message is.
So here is one more question.
Yeah, thanks. A fascinating talk. So do you think that in the first part of your talk, you could make it kind of cell state specific, the modeling, so it would be context specific, and then do something that you did in the latter phase on some style transfer to actually control the regulation?
Yes, the only thing kind of, I guess, challenge with this you need to have a labeled data, and that's not often the case. Especially you can create a labeled data for certain scenario, but not the generalized. With this kind of style transfer, at least with we can generalize across whole protein families, and which is I think what we try to achieve. And it's not. And if you want to improve, for instance, activity, that would be maybe harder. But yeah, there are some ideas, but labels is challenge.
So there's one more question.
Very interesting. Thank you. Did you consider other generative models? I mean, I know you said GAN, you did that for a while ago, but now, I mean, there's been advancements in diffusion models and other generative architectures.
All what I showed now, kind of using all the transformer, now we work with diffusions and so on. So basically, just I didn't show the details how they're under the hood, but all the based on the models that are very challenging to train. Well, the thing is that convolution work nicely with many applications, and we now work with the large language models and diffusion models, and it's a challenge.
But do you see better performance, or they're just too hard to train?
I think it's very hard to say that they are better performing because, as you said, it's just harder to train, and whatever is available out there, I don't think that this is, I would say that is a better model. It's just maybe not trained, under-trained, because if you look at them, we run it with 2 epochs and just publish it with a large data set, and that's it, right? So, I mean, yeah, in academia generally to produce well-trained model is hard because there is no purpose of application, and it it does something and does relatively good, but I don't think that people really spend time on optimizing the resources. It requires a lot of resources.
Thank you.
So we have one final question over there. No, in the back.
Sorry, where? Oh, okay.
So I saw that you have just the active versus not active, but do you look at the structure of the resulting proteins? Do you try to understand why it doesn't work, or is it purely sequence-based? And do you think the structural approach could improve your predictions and transfers?
So I mean, we look at the structures, but we did even some molecular dynamics simulations, and they look fine. So all those structures are fine. I don't think by eye if anyone can tell. I show you the examples, for instance, with this engineered enzyme that at the end with that degrades plastic. I can reveal what's the secret there. It has only one mutation, which basically increased activity over 400 times. And we spend a year doing all sorts of AI and molecular dynamics, microscale length molecular dynamics simulation with 2 people with PhDs and MDs and structural biology. They couldn't figure out why that mutation was doing something. And we make a bunch of experiments. So it's sometimes maybe like quite obvious, like when you're talking about things like binding or something, you can probably interpret it when using structural approach. But when we are talking about catalysis and we, things are getting quantum and so on, I don't think it's really easy to understand.
All right. With that, thank you, Alexei.
Thank you.
And that concludes the scientific session, and I have now the pleasure to introduce Petra Magnusson and Olof Emanuelsson as the DDLS Research School Directors. Most welcome to the stage.
Thank you, Päivi. And we are really happy to be here today to give some information about the DDLS Research School that was introduced by Olli earlier today. And we would like to start by introducing ourselves.
Yeah, so I'm Olof Emanuelsson. I'm an associate professor at the Department of Gene Technology at KTH Royal Institute of Technology. And as a co-director of this research school, we also have
Petra Magnusson from Uppsala University, where I'm running my research group at the Department of Immunology, Genetics and Pathology in vascular biology. That's my focus. And to do this, we are 2, which is great to tango with being 2, but we would not be able to do this on our own. So we have a great team with 2 people working closely with us, and that's Ulrika Wallenqvist and Morskan Seraji, super talented partners with us, and Olli needs no further introduction, I'm sure. And what is it that we are going to do? What is the scope for the research school? It's from university-centered to a national program. And we will do that by selecting the best data-driven life science projects with the best PIs and where there is the best environment for the PhD students. And by doing that, we will also recruit excellent PhD students. And what you heard from Ollie earlier today is that it's been very competitive. This first round of DDLS Research School recruited PhD students with 1% chance to get the position. So we know that they are super talented. And what we're going to do is to educate them and to train them. And train them in activities to prepare them for data-driven life science careers in Sweden. We will provide networking opportunities, which is really relevant and important for them. And we will do that by retreats and national visits. And what exactly more in detail we're going to do, Olof, welcome to present that.
Thank you. Yeah. So we've actually started out today with our first activity. We will come back to that towards the end of this presentation, which, by the way, isn't that far away. We have two core types of activities in the research school: the courses and the annual meetings. Now, the courses, we have now decided about the first year courses starting in 2025. And this is also something that we discussed this morning with the PhD students. There will be a course in open science in the Swedish context. That's going to be a short course. There will be a longer course in deep neural networks with only one E actually in networks for life science applications. And in the fall, there will be a course about principles and tools for fair research practices. And as you all know, I think FAIR stands for findable, accessible, interpretable, and reusable data. The courses for the following years remain to be decided. So this is, in a sense, still work in progress. We also will have an annual meeting, much like this meeting, but focused on the PhD students. So at the conference venue, typically a bit off from the main city, we will have the PhD students for 3 days, 3 to 4 days actually, and we will also invite their supervisors for the first day. And as the DDLS program expands, we will eventually also invite the postdocs of the DDLS postdocs. The annual meeting, I mean, it will look like you would imagine. It will contain, as it says there, workshops, keynote talks, lectures and courses, poster sessions, and of course social events. In addition to the courses and the annual meetings, we will also have a set of other activities, and some examples or suggestions are listed here. This is also work in progress. We do hope to have partnering events with the industry. It's also something that we know from the PhD students that they actually ask for. We will have site visits, possibly summer schools, possibly mentor programs, and also other activities. And this will also be sorted out in discussion with the PhD students. And this morning...
Yes, it all started actually this morning. We had the first gathering, our pre-kick-off for the research school. It was really great to meet these young, talented PhD students. We had a great time together, and we are convinced it's going to be much fun together. And the next event that we will have is the annual meeting, the research school annual meeting in late March next year. So that's when it's going to start. But it's ongoing, and we are super excited, super fun. We're looking forward to what's going to come with these young talents. And
yeah, so I just want to say, so if you are a PhD student, a DDLS PhD student, or if you are the supervisor of a DDLS PhD student, you might want to take note of the dates, actually, 18th to 21st of March, because that has been settled.
Yes, and we're also going to have an event tonight, so it's not over with this morning session. So we will go out and have some social activities, and we have a specific announcement to make
Yeah, so if you are a DDLS PhD student, but you were not present this morning, but you still would like to join for the dinner and social event tonight, then come and see either Petra or myself or Ulrika or Morskan and let us know that you would like to join. And we could most likely arrange with that.
So with that, we would like to thank you for your attention and hand over to the next session, because now it's time for flash talks. And we have a new chair, which will be this session will be moderated by Mariana Pires Braga, DDLS Fellow from SLU, the Swedish University of Agricultural Sciences.
Yeah, exactly.
Thank you, everybody.
Hello, yes, I'm Mariana, and today I'm here representing the poster jury. So we were 4 fellows from the different areas, and our task was to read all the abstracts that were submitted and choose a few to give flash talks. So what's going to happen now is we're going to hear 3 5-minute talks, and there's not going to be any time for questions now, but save your questions and come to their posters, because straight after this, we're going to have poster session. So I think we can kick off with the first speaker, who works at Stockholm University, Inés Cunha, and she's going to talk about interpreting microscopy images with machine learning.
Yes, thank you so much. All right, so as she said, my name is Inés Cunha. I'm a PhD student in Juliette Griffier's lab in Stockholm University, and today I'm here to tell you about how we can interpret microscopy images with machine learning. So, as some of you know, when we use a treatment to treat cancer cells, some of them immediately die from this treatment, but others don't. Others continue proliferating. Oh, sorry, it's supposed to be a video. Other cells continue proliferating or dividing like nothing happened, like the one that you see here in the middle. It continues, it's going to divide, completely ignoring this treatment. And we don't know why. So how do we study why some cells deal so well with treatment and others don't? Well, we do live cell imaging, and in this case, we imaged thousands of cells for over 3 days. We took individual cells and we extracted specific features about these cells, like the signal intensity, the area of the cell, etc., so that our data set now looks more like a tabular data. But there are some limitations to this way of studying cells. And one of them is that we rely on very long imaging experiments, which is obviously very low throughput. And another limitation is that we are the ones that are handpicking the features that we think matter to study the cells, which obviously has a lot of human bias to it. And not only that, but we could potentially be losing some relevant information about the cells by doing this. So here we are proposing a new way of studying live cells. So first we asked ourselves, can we classify or predict the fate of each cell? So given individual cells, can we predict that later on this cell is going to proliferate or if it's not going to proliferate? And then we asked, how early can we classify? Is it that from the very first time point of our experiment, we could already know that later on, days after, this cell was going to proliferate or not? And most importantly, why can we classify? Or what were the visual features within the cell that allow us to know, okay, this cell in 3 days is going to proliferate or die? For instance, is it that cells that were smaller to begin with were less likely to proliferate and cells that were bigger were more likely to proliferate? We don't know, and this is obviously a very simplified example. Well, to answer all of these questions, we could use machine learning, which is particularly good at picking up complex patterns within this complex data type, like images, and in particular, interpretable machine learning to answer why we can classify. All right, so for us to be able to get the essential information that fully represents our image data in a completely unbiased way, we could use autoencoders. And autoencoders are a type of generative AI that take images as inputs and they compress them into a lower dimensional space called the latent space. And then they take only the information in this latent space and decompress them back into an image that should look like the first one. But for it to be able to reconstruct, it's because it contains the essence of our data. It's like if I asked everyone in this room to draw me a picture of a T-Rex, no one's drawing would be the same, but everyone would have the essential features of a T-Rex, like big teeth or like small little hands or whatever. And that's because you have an essential knowledge of what a T-Rex looks like, just like the latent space has an essential knowledge about our data. So we could use this information learned from the latent space to classify our images, so that we are now using the information that's essential about our data in a completely unbiased way. And using these concepts of supervised and unsupervised machine learning concepts together, it's one of the key principles behind interpretability. All right, so in the end, our model was very good at predicting the fate of each cells from the very first time point of our experiments. This means that from the very first minute of our experiments, we could already know with around 80% accuracy what was going to happen later on to the cells. And this has massive implications in terms of throughput, obviously. And this was particularly striking because when we compare the results with what we had with our tabular data, we see that with the tabular data, we really couldn't predict what was going to happen to the cells. And this reassures us that our images had a very important information about our data that we were not considering when we were handpicking the features. Now, because we made our model interpretable, we were also able to extract what were the visual features in the cells that drove the model outcome and what was their biological significance without any a priori knowledge. So if you want to know more about these results and how we built this interpretable model, please come talk to me in poster number 10. Thank you very much.
This is your certificate.
Yeah, thank you.
Thank you, Ines. All right, next up, we're going to hear about Brownian motion data augmentation from Javier Kippen. Welcome.
Okay. Hello, my name is Javier Kippen. I'll talk about Brownian motion data augmentation. I'm doing my PhD in KTH, and I'm working with algorithms and machine learning for different single molecule protein sequencing methods. But today I'm going to talk a bit more about the nanopore field, which is actually a growing field, and it has different applications within DNA sequencing, protein sequencing, and DNA data storage. We can see the publications that have nanopore entitled an abstract that they are increasing every year and reach over more than 3,000. However, the data generated by nanopore sensors is still costly to generate. I'll talk also about a more concrete example that is a data-driven example of a paper published in Nature where they do multiplex single molecule protein sensing with nanopores. And basically, in this Kaiser lab, they have a DNA strand. On one side, they can encode a barcode that indicates like a label for the DNA, and they do that with dumbbell hairpins. And then on the other side, they can design a binding site that can attach for different antibodies. And then when they send this DNA through the nanopore, they can measure with single molecule resolution. So they show in the in the Nature paper that with signal processing methods, they can achieve a high accuracy. But then in a follow-up paper, they show that there's like a neural network that performs actually better for this accuracy. And they call this neural network keeping it. What we were asking ourselves is how can we improve even further these neural networks? So we went back to some other papers in the literature, and there are some simulations where they show how the DNA translocates with physics. And for doing that, they refer to a Langevin equation that basically is a sum of forces here. And you have a driving force that's the electrical field that pushes the DNA through the pore. Then you have some drag forces that are because of the movement in the fluid. And then you have thermal forces that are acting also on the DNA. So we basically took the effects of the thermal forces, and we can emulate some other plausible data that we can generate. So for example, we have ETS, that is our original data, and we can create fake samples with our data augmentation method that we call Brownian motion data augmentation. And with this, we have more data to train our neural networks. And this solves a bit the data scarcity problem that we had with the sensors. So once that we did this, we basically showed that we can improve the accuracy of this neural network that was published. And we also can propose a new neural network that takes the advantage of a better generalization of this and achieve a higher accuracy. So yeah, our data augmentation method is still also simple to apply, and we think it has applications in the whole field of nanopore sensing, but also for other fields like tunnel junctions, which are similar devices of measurement. And we believe that maybe there are other fields that we are not aware of that can also be applied to. If you want to know more, feel free to come to my poster, and thank you for listening. Thank you.
Thanks. All right, and then our last speaker for today is Jujun Lee. And the title of his talk is De Novo Design of Molecular Glues with EvoBind Multimer. Welcome.
Thank you. Good afternoon, everyone. I'm David from Patrick Bryant Group. So while I was making this presentation, I was asking my colleague, so what's the difference, or what's the common thing between snail and human? And my colleague made the joke that, oh, maybe snail doesn't care about mortgage inflation, right? Because they already have a house. Well, joke aside, let's today talk about what's the common between deadly valence and life essential hormones. Well, they're both made of peptides. Nature has mastered the art of creating these powerful molecules, whether it's the most potent venom from, for example, corn snails, or life essential hormones, peptide hormones, for example, insulin, because, for example, insulin can regulate our blood sugar. But what exactly are peptides? Well, think of them as precision engineered messengers. They are chains of amino acids that that are arranged in the specific sequences, and these specific sequences give their unique function, whether they are delivering lethal strike in venom or sending vital signals in our body. Here's the exciting question. Could peptide be a key to the next generation of personalized medicine? Imagine if we can customize a drug for specific diseases or even tailored for individual patients. Here, I present our method, EvoBind 2. This tool can design both linear and cyclic peptide in different lengths based on only target sequence. Unlike other methods, for example, which may require whole structure, the knowledge of interaction domain, our method doesn't need any of this. We let the network decide where the peptide gonna bind. What you need to do, actually, just enter target sequence only and click the button. We already demonstrate that the linear cyclic peptide can be designed in nanomolar affinity in a single shot. We take this technique to another level. Here, I present you another network called EvoBind Multimer. This is our new framework for designing molecular glue, focusing on cyclic peptide that can bridge interaction between the ligase domain and target protein. For example, here, we design a cyclic peptide towards VHL containing ligase and KRAS. KRAS is an oncogenic protein. The design of cyclic peptide is able to bring these two proteins close to proximity for targeted degradation. We also demonstrate the designed cyclic peptide induced KRAS turnover into wet lab. So I think peptide is a current generation of medicine, and it can be designed, it can be customized. To design a peptide binder only from protein sequence sounds interesting. Please come to poster 30. Let's design binders together. Thank you.
Thank you. Thank you. All right, thank you so much to all the three speakers. There were great flash talks. Just before we run away, two important reminders. One, I've been told we start 9 sharp tomorrow morning, so make sure that you're here before that so we can all find our seats. And two, now we have snacks and drinks outside, but don't forget to look at all the amazing posters. That's it. Thank you.